Good afternoon, everyone, and welcome to day two of the Global Resilience Summit 2026 Mental Health, AI, Financial, and Technological Resilience in Medicine and Biomedical Sciences. Yesterday, we had a productive closed door conference, day one, with University of Colorado, Colorado Springs, on mental health resilience. The sessions featured insightful talks by Dr. Lauren Shaw, Dr. Charles Benight, and Dr. Nicole Weis. Today's sessions bring together perspective across AI, neuroscience, biotechnology, and healthcare. This is going live on YouTube, by the way. We will begin with the inaugural address by Dr. Sukant. We have Dr. Sukant with us. He is a scientist, entrepreneur, and founder of Cure Group. He is working at the intersection of AI, neuroscience, healthcare, and education. His work spans areas such as AI in drug discovery, neuroscience, and computational approaches to complex biological systems. The stage is yours, Dr. Sukant. Welcome.
Okay, thank you, Zina, for the introduction. So, I will say only a few things, but they're around the basic idea of a phrase called riding the tiger. In this age, unless you are close to retirement, you got everything sorted. There is absolutely nothing that can really give you a sense of security. I'll not say certainty. This world is increasingly uncertain with global conflicts, with global mobility being altered. You see changes in visa, you see regimes collapsing, things which appear to be very stable, very big, you don't know. The game of superpowers also you don't know. Similarly, there are so many firms working on different areas, and then there are companies raising successfully, not claiming, successfully raising money for solving all diseases. There are companies which are doing that. There are companies raising money in billions on longevity. There are AI companies with valuations unheard of previously in hundreds of billions. What can you do in this world which is increasingly at one end filled with rosy picture of hundreds of billions being raised or insane money being made in stock market, and the other end there are MBBS students, PhDs, writing and delivering food. I don't want to horrify you on the number of people doing small jobs at Swiggy, Zomato, all these things, platforms. So the world is increasingly of two halves, and actually, this current revolution is hurting the AI have nots in white collar. So you have two options: either live in the basement of your mom, so to say, retire early, live frugally, um, or if you're super lucky, find a government job in a semi-socialist country like India, where a lot of our audiences are from, which requires connections more than merit, um, even at the level of so-called top research institutes and whatnot. Um, or alternatively, ride the tiger. I am going to preach what I practice. Very bad at preaching what I don't practice. For example, I can't be giving you too much of advice on eating healthy. The people on this call who have dined with me and they know how much I gorge on fast foods and all, or other things. What I can give you honest advice is on riding the tiger. Riding the tiger phrase means capturing the frontier spirit, right? So, can you be at the frontier end of AI? And when I say frontier end of AI, it's not just AI. You cannot be at the frontiers if you don't have a wide natural intelligence. What I mean by that, if you're still calling yourself just an immunogeneticist, or a genomics person or a microbiologist, and you're still young, you don't have job security, you've lost the plot. Innovation, true innovation, as I again said, unless you have a government security of some job, you're a lucky one. Whether you're got it fairly or not, that's up to you. But if your goal is to innovate, and when I mean innovate is not some five papers with in a journal above impact factor 10. When I'm saying innovate means making a real difference and actually products out in market, services out in market, some tangibles, right? So if your goal is to do that in the current world, in a hyper competitive world, you face several challenges. A lot of competition. The barrier to build something has gotten easier, not as easy as someone on X, formerly Twitter, would make you believe, but it has gotten easier. The other thing that you're facing is there's a lot of noise, real as well as fake innovation, too much competition. Third, you are dealing with a lot of people who are anxious, right? Whether they are your friends, they are your clients, they are your employees, they are your family members, there is an uptick in anxiety and there's reshuffling of global order. Within this, what is the way? I don't want to be a prophet of anything, but I'm telling you few simple principles. Number one, integrate across domains. Okay, as many domains as you can, read widely. Um, your typical topper of the class who just reads few subjects does very well in exams. Hardly any one of the people who have been self-made billionaires are those. True intelligence to integrate across domains. Number two, learn to question authority smartly. When I mean smartly is, I'm not expecting you to go on protests. If you're going in too many protests, take it from me, you're not going to make it big. If you have in past, change that. When I say find your place, right? Let's say if you say that you're in some system in some hill state and the best engineering college offers this incubation and will charge you this much money and that's all you got, maybe you have circumstances which bind you, or have a back, have a spine and say, no, I'll have the courage to take few failures and be in a place which accommodates that. I'll question the authority in a right way, respectful way, but have a spine to believe in yourself, spine to believe in the future. Next thing is pick your battles. Learn to say no to 90% of your battles. Is it needed? Don't get me wrong, I had so many bad experiences in India, Indian science, and some in New York. Texas was mostly good for me. My collaborators were extremely good at NYU, Northwestern. Um, but I am fighting selective fights. And anyone who knows me, it's not the reason that I'm weak. I would not like to, on a YouTube broadcast, tell you how many top science professionals have been fired because of my report. Ask anyone in science, they'll tell you the number is very high. But I end up saying no to 90 to 99% of fights. And I'm also gradually learning to say no to 90 to 99% offers. I'll give you an example of Pepsi Vice President, ex Vice President Daniel Nelly, who'll soon be presenting, not in this conference, next or the next to next, offered me to tie him to some hospital to supply some healthcare thing. I said I'll introduce him. He was offering commission. I said no, that adds responsibility. I don't have time. Time for me, you may think you have time if you're half my age in early 20s. Um, but trust me, no one has too much time. The sooner you learn to say no to 90% or 99% people, in fact, if someone is not performing in your group, learning to say no to them politely, that this is your time is up, move on, find something else. Learning to say no is going to be your superpower in the times to come. Okay. Then, when I said choose the right environment, means surround yourself with the right people and right geography, if that's possible, otherwise virtually. Lastly, what I mean by riding the tiger is knowing AI in two ways. Okay. And before I get to the AI point, I'll talk about three other things of riding the tiger. One is learning to take responsibility of not just your actions, but your team's. Okay. Something goes wrong, I can yell at my team, but being the CEO, at the end, if the company goes south, it's my responsibility. I shouldn't be CEO if I'm the weak CEO. And I can't correct, course correct, take the right decisions, right? I'm not sorry for being a bit blasphemous, prime minister of some country or member of some committee, right? If I have formed something through my blood and sweat, I need to be strong enough to take whatever decisions are needed and be prepared to take, like, bear the cost of it. Because it's not sort of socialism, it's not someone else's money, right? If I'm making it, I'm making it. If I am losing it, I'm losing my own thing. So, willing to take responsibility of my team at the end of the day. And I'm not trying to say anything unique about myself. That's the mantra for anyone. Whether you're working for someone, if you take responsibility of even three people, see how much more resilient you will be in the times to come. Okay. It doesn't require a formal MBA. Secondly, taking initiative. Thirdly, asking a smart person for advice constantly, knowing that we're all humans. At least I know for sure none of us is chosen one, none of us is Messiah. Uh, so we'll all make mistakes. In fact, every day I look back and I can think of counting five, six mistakes I made the previous day. The goal should be to not repeat the same mistakes. The goal should be to talk to someone smarter than you. That gets me to AI. Is AI truly AGI? The answer is yes and no, and I'll not go too much into the AGI part. I'll tell you, we are also working on some aspects of superintelligence. We are trying to work through three different novel means. One of them is instead of using integrate and fire neurons, which every bit of neural networks are made of, we're working on different kinds of neural architectures, neural computations, including inhibition in the circuitry. Secondly, instead of computation being centralized, where motives are derived from human brain, we are working on distributed computing. We call it Octonet, inspired by Octopus. Thirdly, we are working on other aspects of computation inspired by non-brain intelligence, such as plants, fungi, ecological computing. So we are also working towards superintelligence. The answer to is AGI is there or not? Yes and no. If you believe what Mark Anderson says, the answer is yes. In fact, what even Crusoe said. These are great thinkers. People I have a lot of respect for. Uh, I would say the best scholars that are out there, despite you may try to say Crusoe, obviously, no one will contest, with Mark Anderson being a top Trump AI advisor and being an investor. People would say investor versus professor. He's a lot smarter than any of the top professors I've worked with. I've worked with the best of the best in mathematics. In fact, I collaborated with a top guy at NYU during my PhD mathematics part. So if you believe what is there, but I can get into technicalities and they will be slightly besides the point, because are transformers capable of superintelligence? Is language inherently having logic to it, or are transformers having capacity beyond what we think? Because mostly what you're talking to are transformers. Instead of simple probability of one word another after another, you're increasingly talking to models which are talking of probabilities between different concepts and not just some lines or words or phrases. So we are not at the AGI in terms of Terminator AGI, but we are definitely seeing what is in loose terms called AGI walk on its course. It'll soon start walking like a toddler. So we are at the beginning of dawn, and you need to either decide to ride the tiger. So the first advice that I'm giving you is talk to AI at least every day. If you have a good friend, I don't mean by good friend someone who drinks beer with you or waste your time, if you have a good friend, do talk to them also. Don't cut out your circle. AI does make mistake, and AI is only as smart as you are in some ways. Treat it as Google, it'll be like a Google. It'll give you answers, and sometimes it'll try to please you and give you wrong answers, not just hallucination. Okay, because there is a commercial interest in AI company wanting you to keep using its AI. Use it as a smart person you will even argue with. Ask further questions when you say this doesn't make sense. Like yesterday, I was talking to Gemini and discussing are people, let's say my father's side of heritage, someone with surname like Kurana, are they closer to Europeans or are they closer to Tamils? And the answer was closer to Europeans. When I started doing maths, it started giving me different answers. And I started asking it assumptions. While the initial answer was correct, that people from Punjab are probably genetically close, much closer to Middle Easterns and Europeans than they are to Tamilians. The initial estimate was much higher than when you start asking its assumptions, right? What are you comparing where, right? Because obviously the baseline is that all humans are very similar. What is the point of split? What have you considered? Are you considering 120 years, 120K years point of split from African population or 70K? You start asking it questions. Moment AI is forced to think correct, and you say, what were the assumptions of this paper? Are you just regurgitating the things, right? You start getting smarter answers. Now, imagine if you had just treated it as a Google and relied on first answer versus the very detailed answer I got, and I kind of reached the end of AI where Gemini said that this is the current state of knowledge, right? So to say, I broke Gemini. You can also, right? So it's not like something I've done is extraordinary, basically, but I got Gemini to say this is as much as it knows. So, but you have to be smart, right? So talk to AI. Second part of AI is learn AI tools in two professional categories. One is how to draft things better, how to make better figures, how to write better emails, all such things. Typical corporate job, any job that you need. Second is your actual skills in medical field or biomedical field or pharma or biotech. Ride the tiger, and if you really ride the tiger very well, then break free in the right geographies, do innovative, do crazy things. Um, in this inaugural lecture, I would like to get to the end, which is sort of the beginning also of announcing that soon we'll reach a stage where we will become a necessary daily dose, not a weekly dose, daily dose for anyone in life science, medicine, pharma, biotech. There will be conferences organized on a daily basis, 365 days a year. And so that the evening live broadcast becomes part of everyone's necessary appetite, necessary dose to succeed in life science. That's the mission. The way we are structuring Healers Harbor, my vision is to make it go live on 90% plus things. The reason to do it is we want it as a great tool for education, empowerment, and also publicity. Initially, we aren't reaching out to partners. Later, we will develop business concept around mass reach. We also have something incredible which you guys may not have been exposed to. Is my engineering team, which I'm very proud of, Arsh, Sahil, Aditya, and all. They have developed back-end networking model, which I believe will be soon better than Facebook or LinkedIn, but only for professional biomedical purposes. There exists another website of Healers Harbor that you're not aware of and will be unfolded and uh, revealed to all of you and to everyone in next 2 3 months. So we are building something great, and I'm really thankful for everyone to join this great journey together, and thankful for the opportunity to give this inaugural lecture of day two. Thank you, everyone. Happy to take questions.
Thank you, Dr. Sukant. So, do we have any question you can write in the chat? For those watching on YouTube, you can also write your questions on the chat. There's no question as of now, so I think we can proceed, sir.
Okay, so let's move on to the next talk.
Okay, there is
Uh,
a question from Dr. Abhijit.
yeah,
He
good
has,
question. Abhijit is a good friend. I'm sure whatever question he has is it's there to stump me, right? What is the question?
the question is, what kind of agentic AI is required for biological data analysis?
Okay, so one question at a time. So it's later. So yeah, Abhijit, I think so. Agentic AI, the best purpose of agentic AI is to not be the next AI tool itself. The best purpose of agentic AI is to essentially find the right tools and lay them in the order, right? For example, the bioinformatic framework which we have, right, which we have even, I'm assuming has been even sent to you for Parkinson's. Yeah, I did something, so I'm sure that I've sent to you with 30 plus tools in a rigorous way, around 70 tools. The right use of agentic AI, and doesn't have to necessarily be, you know, something from Clawbot or some other thing. It can be even the standard agentic AI that Google is offering, in fact, use for free. If you're missing out, you can even register for things for tomorrow. Tomorrow is the deadline. I can send you the message. For the first round, I'm sure there'll be multiple rounds and not just from Google, everyone. So the purpose of agentic AI is to essentially have the agency which humans have, right? So giving the end purpose to the agent. The end purpose, instead of what we were used to, Abhijit, you and me, including in our combined work or our separate work, we were looking at, let's say, drug discovery from 7, 8 conceptual frameworks. Number one, let's say we are looking at what is binding to what, right? Molecular docking, MDS. Then we are looking at the molecule doing fragment analysis, QSAR. Then we are looking at the standard EDMET toxicology, especially absorption, digestion, excretion, metabolism, toxicology. Very rarely with AI, people are also doing things with other aspects of sutex, kinetics, and formulation. And then looking at epigenomics, then some people were, you and I were in that limited category of looking at it evolutionarily and systems pharmacology approach. And then eventually combining all of these. The purpose of agentic AI is to not be one tool. Even in each category, there are 10, 20 good tools. Right? If you integrate genomics, there itself probably 40, 50 tools. The idea is to agentic AI, just like humans, have agency. So the most advanced agentic AI you can use is to have something which is doing all of it, but unlike you and me, not in your case, not in my case, needing, let's say, a cigarette break. In my case, needing a lot of kebab breaks and, Diet Coke breaks and sleeping. So agentic AI doesn't need to sleep, right? Tools don't need to sleep. So how can they increase productivity? Have agency, what the word agentic AI is. So I'm sorry, I'm emphasizing the very basics of what is agentic AI. And the reason to emphasize that is that people who are not even from AI background understand, and they also get the basic principle that agentic AI helps you to organize things, to use other tools. And that's what you need, essentially a pipeline, but not a static pipeline. If something is wrong, not just that output of this goes to this, right? Understanding, even asking questions to some level, right? What a human does. And the more advanced the agentic AI is, the more it can replace humans. Does that answer the question, or would you like a more less conceptual and more technical answer? Okay. So what was Sohit's question?
So his question is that you do you agree AI could easily replace research interns or assistants altogether?
AI can easily replace professors. What do you mean interns?
Right. That sets the context for the discussions ahead. Thank you, Dr. Sukant.
Okay,
There are
thank
no
you.
more questions. Yeah. Now, it is my pleasure to introduce Dr. Theodore Tursky's talk. He's a research scientist at the Harvard University. His work sits at the intersection of neuroscience and social context, exploring how environmental and socioeconomic factors influence brain development. So, in this session, he'll be talking about relating poverty to brain structure, offering important insights into how lived experiences shape neurological outcomes. So, let us move to his presentation now.
Good morning, good afternoon, good evening, depending on when you're watching this recording. My name is Ted Turesky. I'm a staff scientist at the Harvard Graduate School of Education. And prior to that, I was a postdoc at Boston Children's Hospital, where I conducted, along with collaborators and other members of our team, this work that I'll be presenting today. Before I go any further, though, I should state that I have no conflict of interest to report, no conflicts of interest to report, financial or otherwise. So what I'm going to be speaking about today is this relationship between poverty or the associated biological and psychosocial risk factors and brain structure and function. And this study was conducted in infants who were growing up in conditions of extreme poverty in Dhaka, Bangladesh. So before we go into the neuroscience, just wanted to zoom out a little bit and talk about the neurocognitive deficits with which child poverty is associated. And so these deficits are myriad, and they can transpire and manifest at different times over the course of development and later in life. So some of the examples of deficits are in terms of cognitive control, in terms of working memory or language, which seems to be the most substantial based on this meta-analysis of effect sizes, showing the greatest deficit in language over multiple studies. These deficits also include diminished academic performance and mental health problems. But there's a big gap between childhood poverty, which one can measure with income to needs, which is the overall household income divided by the number of household members, and various cognitive impairments. And so presumably, if one experiences a cognitive impairment, there has to be something going on in the brain that gives rise to that. And so that could be in terms of gray matter, white matter, or activation measured with fMRI, or functional connectivity also measured with fMRI. And we can talk about what functional connectivity is a little bit later for those unfamiliar with the term. Another thing to keep in mind is that poverty doesn't affect the brain directly, but it does so through various risk factors. And those risk factors could be biological or psychosocial. And these are just some examples. And we're going to be talking about some of these: inflammation, malnutrition, and stress. We probably won't talk about reduced language input too much. So one study that kind of examined this pathway, we'll talk about the biological risk factors first, it was also conducted in infants in Bangladesh at 18 weeks of age, when they measured concentration of this particular inflammatory marker. And then they looked at the cognitive outcomes, cognition score, in toddlerhood a couple of years later. So this is also, and this over here on this graph is a language score. You can see that the effect sizes aren't particularly robust, but the effect seems to be reliable overall. But this didn't look specifically at the brain. So these were measures of systemic inflammation, and then the cognitive outcomes. So looking at more specifically at the brain, this study examined inflammation, which was measured from a mother's amniotic fluid in children who experienced or manifested these periventricular white matter lesions. It's the plus sign, or they did not. And you can see that overall, the kids who did not have the periventricular lesions had inflammatory marker concentration, IL-1 beta in this case, close to zero or very low at least. And whereas the children who did have the periventricular lesions, a lot of them had higher concentration of, well, a lot of them relative to proportionally compared to the kids without the periventricular lesions had a wider array of inflammatory marker concentration. So overall, there's not strong evidence with the inflammation and the link between inflammation and brain structure that I've shown so far, but I think this study presents a little bit more compelling evidence. So this was an animal study in which they injected inflammatory cytokine into mice to induce systemic inflammation, and then they down the road perform this diffusion imaging, which we can do in humans too, but those data have yet to be analyzed. And so fractional anisotropy, which is a measure of white matter organization in the brain, is modeled here on the y-axis, and the different white matter, canonical white matter pathways are on the x-axis. So CC is corpus callosum, Sing is cingulum, EC is external capsule. And so you can see that for all of these tracks, the mice who received the cytokine injection had lower levels of white matter organization. And so the study then tried to figure out the mechanism that might be leading from systemic inflammation to lower white matter organization. And what they observed was that the mice with cytokine had lower myelination, and that a possible reason for this was that the inflammatory cytokine was disrupting oligodendrocyte maturation. Just a reminder, oligodendrocytes are what help generate the myelination of the axon. And so bringing this all together, after the diffusion, the mice were tested for long-term memory, and those who received the cytokine performed worse overall. Malnutrition is another risk factor, one that's very common in conditions of extreme poverty, and malnutrition has been linked to reductions in cortical cell size and a number of other cellular properties. And one thing that's important to mention is that inflammation and malnutrition are not totally independent. They sort of feed off each other. They have a reciprocal relationship. For instance, usually the inflammation is an indication of some underlying infection. And if you are in that condition, it's less likely that you're going to be reaping all the nutrients from the food that you're eating, if you're even eating at all during that illness, that period of illness. And the relationship goes the other way as well. There have been studies that have articulated this, investigated this more thoroughly than I'm explaining here. Another thing to keep in mind with malnutrition is it doesn't just affect the brain, it also affects the rest of the body, and that can manifest in a few different ways, one of which is stunting. And so in this picture, this child in the middle is the same age as the other two children in the photo. However, she is significantly smaller, and she is experiencing stunting. And stunting is something that's very common around the world, particularly in Southern Asia and Sub-Saharan Africa, where the rates of stunting are quite high in children under 5. I think it's gotten a little bit better since 2017, but this was the case, at least then, and it still is not ideal. So one thing about stunting, which is one of 3 main anthropometric growth indicators that are used globally often by UNICEF and the World Health Organization, is that it indicates it can be a proxy for inflammation and malnutrition. It's often used that way too. So if the 3 measures that are going to be considered as anthropometric growth measures are stunting, underweight, which is just low weight for age, and wasting, which is a sort of proportion of weight to height. So stunting, height for age, underweight, weight for age, wasting, weight for height. And so these diminished anthropometric growth indicators have also been linked with cognitive impairments. And so what we're thinking is that since these are proxies for inflammation and malnutrition, we can kind of get at this question of, get at this relationship between biological risk factors and brain alterations by examining links potentially between diminished anthropometric growth and gray matter structure and white matter structure in particular. We're not going to look at the relationship between anthropometric growth and any of the fMRI measures at this point. I think from a hypothesis perspective or from a theoretical framework, it makes less sense. So the participants that we had were 29 infants, and these are their height for age scores. You can see it's quite low. These are standardized measures, but it's quite low compared to the others. I mean, also if it's below 1, it's below 2, it's an indication of stunting. And so this is just the average, but it's so there are a number of kids who are officially stunted in the sample. Weight for age and weight for height. And this is the income to needs amount. It's in Bangladeshi taka currency. And you could see that years of maternal education, the mothers don't have too much of an educational background. And this is another measure that's often used in the context of poverty or studies of socioeconomic status. Something methodological to keep in mind is that when you're examining MRI data from infants, there's this interesting thing that's occurring where, because there's very little myelination in the early months of infancy, in the early postnatal months, there's very little white matter actually in the brain. And it often appears darker than the gray matter. So you can see there's some myelination going on in the more medial areas, but most of the axons over here on the periphery are unmyelinated. So the quote white matter appears darker than the gray matter. So this challenges and stymies all these segmentation tools that are designed and tested on adults. And so we have to use specialized tools. And even after that, the segmentation, the labeling of gray matter voxels and white matter voxels is not particularly accurate. But we do the best we can. And we had a pediatric neuroradiologist do the ratings afterwards. So after the segmentation process, we essentially just summed up the voxels that were labeled as gray matter and the voxels that were labeled as white matter in these infants and tested for partial correlations between these brain measures and the anthropometric indicators, HAZ, which is stunting, WAZ, underweight, an indication of underweight, and WHC, which is wasting. So overall, the relationship between the anthropometric measures and gray matter volume, we didn't see anything particularly robust. There was the significant, sort of uncorrected, significant relationship between HAZ and gray matter volume, but as I said, it's uncorrected, and so we don't put much weight on this. Another thing to keep in mind is that the structural growth is very different for males and females, so we separated them when conducting these correlations. It's the main reason for using semi-partial correlations, or at least partial correlations. In contrast to the gray matter results, the white matter volume seemed to exhibit significant relationships with HAZ and WZ, with the stunting measure and the underweight measure. So, interim summary, HAZ and WAZ were correlated with total white matter volume in these infants growing up in extreme poverty. And so if we assume that HAZ and WAZ can be used as proxies for inflammation and malnutrition, then maybe those two biological risk factors could explain this relationship that we're observing. So we've established this green pathway here, and now we'll move on to the psychosocial risk factors. So when considering psychosocial risk factors, one important area in the brain to consider is the amygdala because of its projections and modulations of the hypothalamic pituitary axis, pictured here. And so it projects to the hypothalamus, and it has glucocorticoid receptors that provide this feedback loop. And actually, other studies have examined the amygdala also in this context of stress. And this was a study conducted in infants in the US, not infants growing up in extreme poverty elsewhere in the world. And they looked at functional connectivity, which is really just a measure of the temporal correlation of activity between two areas. So one area varies, maybe it's like in its activity like this, and if another area has activity that's kind of similar, oscillates similarly in sync, then we would say those two areas are functionally connected. It's possible that one area's activity is like this, and another area's activity is like this. Those probably not functionally connected. So what this study examined is this functional connectivity between a region of interest, posterior cingulate cortex, in the posterior lobe, medial posterior lobe, and they looked to see which other areas in the brain exhibited functional connectivity that was correlated with their measure of early life stress. And what they found was that there was this functional connectivity between this seed region and the amygdala and also the medial prefrontal cortex that was correlated with early life stress. And interestingly, they also brought in some outcomes they measured later on, and they showed that this functional connectivity between the posterior cingulate cortex and the amygdala was in turn correlated with negative emotionality outcomes later on. So maybe something going on with the amygdala early in life here. This study put the seed region this time in the amygdala and looked for other areas that exhibited functional connectivity with the amygdala in a way that was correlated with income to needs, which again is a measure of socioeconomic status, or in the extreme case, a proxy for poverty. And what they found was a negative correlation here. And that makes sense. The other one was the one I showed on the previous page was, I think, also a correlation. I can't remember the direction. But as income to needs goes up, the functional connectivity between the amygdala and precuneus goes down. And that makes sense. These things are part of different networks, and so it makes sense that they'd be, you'd want them to probably in a more typical developmental case to be more segregated and to not exhibit this functional connectivity. So it was a negative correlation here. So we thought, okay, the amygdala makes sense to investigate in the context of our study, which is a case of more extreme stress. And so we took the participants that we had in the prior study. They also had functional connectivity, or they also had fMRI data that we could use to generate functional connectivity measures. We divided them into children below the poverty line, which was a measure provided by the World Bank, and above the poverty line. I mean, you can see either way there, the income to needs measure is low. The maternal education measure is low. But this poverty line provided an objective dividing point. And before we looked exactly at the relationship between the amygdala, functional connectivity, and stress or other psychosocial measures, these are just showing the overall connectivity that different areas have with other parts of the brain. So if we put the seeds in the amygdala, then there are areas that are around other voxels around the amygdala that are also correlated with it. So this is not extending too far from it's sort of the local area in this network, but let's see, and the same with the visual cortex. If we place seeds in the calcarine cortex, we see the visual network and the other voxels that are part of that network. Similarly with the auditory network and the motor network, although with the motor network, when we put, because the motor system is so dispersed, if we put seeds in the precentral gyrus, we see that there are areas in the cerebellum that are also functionally connected to those precentral gyrus areas. But again, we're just focused right now, based on our hypothesis, on the amygdala. And so what we find is that there is actually this association between the amygdala, functional connectivity between the amygdala and precuneus, which is pretty similar to the areas that were shown in previous pictures. That functional connectivity is correlated negatively with income to needs. It's correlated, just like in the previous study, it's correlated negatively with maternal education, and it's correlated positively with the family conflict score, which is another indication of stress. So this makes sense. The income to needs goes up. The functional connectivity between those areas goes down. And as the family conflict goes up, which itself is negatively correlated with income to needs, that the functional connectivity would go from less negative to either no functional connectivity or slightly positive. So in summary, the relationship between poverty and brain function is detectable even by 2 months following birth. So remember, these kids are very young, but this relationship seems to be somewhat persistent regardless of the population that's examined. This is bringing in the anthropometric indicators, which we did, I think, test a little bit. We didn't find any correlation between that and amygdala and precuneus functional connectivity. This is just to show that the anthropometric indicators are sort of affecting structure, but the more psychosocial factors seem to be more affecting the measures of functional connectivity. Lower income to needs and greater family conflict, which are themselves sort of antagonistic, if you could put it that way, with greater or associated with greater connectivity between the amygdala and precuneus. And this sort of fits with this mediation model of income to needs going to amygdala precuneus or amygdala lingual gyrus in this case, and some outcomes later on. Okay, so this is a summary of the findings that we had from this study, both with the biological factors and the psychosocial factors. And these are all the people I'd like to thank in their respective institutions. Okay.
Okay. So that offers an important perspective on the relationship between poverty and brain structure, right? Now we are moving to next presentation by Dr. Shikha Agarwal. She is a senior researcher at IonCure, and she is working at the intersection of AI and pharmacological systems.
Hello everyone, I'm Dr. Shikha Agrawal from IonCure and I'm honored to present the convergence of artificial intelligence and neuropharmacology, a new era for Alzheimer's therapeutics, where we will examine the paradigm shift from labor-intensive research models to the AI-driven pipelines that are successfully advancing a new generation of Alzheimer's drug candidates into clinical trials. Let's dive into the topic. For decades, the search for an effective Alzheimer's treatment has been one of the most difficult journeys in medicine, marked by a staggering clinical failure rate of over 99%. This was largely because we lacked a deep understanding of the disease's complex multifactorial nature. But today, We are witnessing a fundamental shift by moving away from labor intensive traditional models and embracing an integrated data driven approach powered by AI. We are finally addressing the bottlenecks that held us back from identifying biological targets to predicting how well a drug can cross the blood brain barrier. Let's look at how we are now designing entirely new molecules from scratch using these tools. One of the most advanced applications of AI is generative chemistry, which allows us to explore a chemical space of 10 to the power 60 potential molecules to find the ones most likely to succeed. A landmark example is DSP-0038, the world's first AI-designed Alzheimer's drug to enter clinical trials. It targets Alzheimer's psychosis by acting on two specific serotonin receptors simultaneously, a dual target feat that traditional discovery often struggles to achieve without causing motor side effects. Similarly, ISM 8969 was designed by in silico medicine to inhibit the NLRP3 inflammasome, a key driver of neuroinflammation. What's truly remarkable is the speed. AI shortened the discovery timeline from the traditional 4.5 years to just 1.5 years. But discovery isn't just about creating new molecules. It's also about finding new users for the ones we already have. Yes, I'm talking about drug repurposing. Drug repurposing is an attractive shortcut but because these compounds are already proven safe for human use. AI has turned this from a game of chance into a systematic science. Take sildenafil for instance. Using network medicine, researchers identified it as a top candidate that interacts closely with Alzheimer's genomic networks. This hypothesis was backed by real world data showing that users were 69% less likely to develop the disease. Then we have bumetanide, a diuretic that AI identified for its ability to flip the transcriptomic signature of the disease. It is currently in phase 2A trials to see if restoring neuronal chloride balance can slow down cognitive symptoms. Moving on to the next slide. Beyond identifying molecules, AI is also optimizing the clinical trials themselves to ensure they are more efficient and accurate. AI is now essential co-pilot in the development pipeline. For example, IGC Pharma uses AI platform called Agentic Harmonization Assistant to harmonize fragmented data across studies, reducing months of preparation into mere days. They even use geofencing to accelerate patient recruitment in underserved communities. In the phase 3 arena, we have AR1001, an oral therapy with triple action mechanism that protects neurons while clearing amyloid and tau. Furthermore, we are now using digital biomarkers like modality.ai to capture speed changes and emerald wireless sensors to track walking and sleeping patterns in a patient's home. These tools gives us a continuous real-world look at how a drug is actually working. As we look toward the future, the focus is shifting from single drug solution to complex combinations. Because Alzheimer's is so complex, the future likely lies in combination therapies that target multiple pathways at once, like neuroinflammation, metabolism, and protein clumping. AI frameworks like DeepDrug are now used to identify most synergistic drug pairings. We are moving toward precision neuromedicine, where AI will help us match a patient's specific genetic and biological profile to the drug most likely to work for their unique subtype of the disease. In conclusion, we are standing at a major turning point. The integration of AI into Alzheimer's research has evolved from a simple screening tool into a comprehensive framework for development. While the challenge of achieving full disease modification is still significant, the foundation we have built from discovery speed to trial optimization is robust. We are finally moving away from black box of the past and into a data-driven era where a cure is no longer just a hope, but a calculated objective. Thank you for your time.
Thank you, Dr. Shikha. Our next speaker is Dr. Jyotsna Polyedat from JPR University, Chennai, with expertise in nanotechnology and applied AI. She will be speaking on AI-driven design and optimization of nanomaterials for water treatment.
Hello everyone, I am Dr. Jyotsna Polyedat from JPR University, Chennai, India. So today I'm going to present about one interesting topic, that is AI-driven design and optimization of nanomaterials for water treatment applications. So in this So we know that water pollution is a serious global concern caused by the contaminants such as heavy metals, organic pollutants, inorganic pollutants, pharmaceutical, microorganism, etc., which can pose risk to both human health as well as the environment. So traditional water treatment methods are not fully effective because they often involve high operational costs, lower efficiency, and poor selectivity towards some specific pollutants. But nanomaterials have emerged as a promising solution due to their unique properties such as high surface area, adjustable surface chemistry, and improved reactivity, which make them highly efficient in removing contaminants. And these materials are widely used in like key process like adsorption, catalytic degradation, antimicrobial treatment, enhancing water purification performance, etc. So AI like plays a crucial role by accelerating the discovery and this optimization of nanomaterials by reducing the dependence on this trial and error methods. So these techniques can accurately predict the important properties such as absorption capacity, the efficiency of the catalytic properties, and any antimicrobial activities, degradation process, etc. So overall, the integration of AI with this nanotechnology significantly reduces the experimental time as well as the cost, while improving the efficiency and effectiveness of the water treatment systems. So nanomaterials such as oxides, metal oxides like carbon-based nanostructures or even hybrid composites have shown a significant potential of water treatment applications. But however, their full potential is not yet completely utilized. One of the major challenges is that the selection of appropriate nanomaterial and in the methods we use, like trial and error methods, which is time consuming and inefficient. So additionally, the performance of these materials, nanomaterials, especially in adsorption and degradation processes, highly sensitive to synthesis conditions such as temperature, preparation methods, pH values. So another limitation is like lack of like the predictive models to clearly understand and forecast the interaction between the pollutants as well as the nanomaterials. So to address these challenges, this particular topic introduces an AI integrated framework which can enable the intelligent design and optimization of nanomaterials together. So this approach helps in developing efficient, targeted nanomaterials specifically tailored to remove the particular water contaminants or pollutants, which improve the overall treatment process. So the first objective is to develop AI-based models that can accurately predict the adsorption efficiency of different nanomaterials, helping to identify the most effective materials without an extensive experimentation. And second objectives focuses on optimizing synthesis conditions such as different parameters, pH, temperature, dosage, to achieve the maximum removal of the pollutants from water. Another objective is to design the multifunctional nanomaterials that can simultaneously perform adsorption. It can exhibit antimicrobial properties. It can enable photocatalytic degradation of the contaminants. And finally, the study basically aims to build a data driven decision making system that integrates all insight to support the selection and design of advanced water purification technologies. So nanomaterials can be different kind of nanomaterials. So one form of nanomaterials which is very efficient for the water treatment is metal oxide nanoparticles, like zinc oxide and titanium oxide. So these forms of oxide nanoparticles can improve the photocatalytic degradation methods. So these are the examples, that is ZNO, TiO2, which can be used for photocatalytic degradation of dyes as well as organics, and magnetic separation of heavy materials. So this is also another method that usually uses for the pollutant removal purposes for wastewater treatment, and copper oxide is mainly used for the antimicrobial activities. So this explains how the photocatalytic purification works. Like we have different steps that we will utilize for the photocatalytic degradation methods, and in which we will be exposing our for solution as well as the nanomaterials included to the UV radiations, and there will be an electron hole as well as the gaps left after the electrons. You can see that this is electron excitation, and this electron is excited to the conduction band, and which will turn into a hexavalent chromium as well as a chromium oxide. And what happens, there is an oxidation process occurs here, and what happens, it will converts into a superoxide anion, and in which reactive oxygen species will purify the toxic substance, and thereby you can remove the pollutants from the nanoparticles by metal oxides. Another is carbon based nanomaterials. So carbon based nanomaterials, so the one major important what do you call peculiarity of carbon nanomaterials is its adsorption capacity. So here what happens, adsorption is a little bit different from absorption, in which in nanomaterials you will have different active sites in which the pollutants will come and adsorb above the surface. So thereby you can, if you do the filtration method, you can filter out the pollutants as well as the any contaminants. So carbon nanotubes is one of the most and fast pollutant transport for the removal, and activated carbon nanocomposites, through that also you can have the effective purification. Another one is nanocomposite, that is hybrid structure combination, combine adsorption plus catalytic plus antimicrobial properties. For example, the nanocomposite of silver, zinc oxide, and graphene composites plus iron, and these two combined together, so they can have combined efficiencies together, which will be more effective for the water purification method. So these are the basic nanomaterials which we can prepare for the wastewater treatment. Now comes the role of artificial intelligence in water treatment nanomaterials. So for the photocatalytic efficiency, the basic models, AI can predict the photocatalytic degradation efficiency of the pollutant. So AI models are used to predict how efficiently a photocatalytic can degrade the pollutants under different conditions by reducing the need for extensive experiments. So important factors such as band gaps, surface area, light intensity, and catalytic composition are analyzed to understand their impact on photocatalytic performances. So artificial neural networks, that is ANN, it is a complex model and nonlinear relationship between the input parameters like band gap. If you're fixing a parameters like band gap, pH, or light intensity, these two can combine together to get an output of degradation efficiency. So it actually mimics the, like, the learned patterns from data and provide an accurate predictions. So random forest and XG boost, so these are on some machine learning models that combine multiple decision trees to improve the prediction accuracy. So they also help identify which factors are most strongly influencing the photocatalytic efficiency. And support vector machines, that is, it is used for classification and regression tasks. So it helps to classify the catalyst as high or low efficiency, and can also predict the degradation performances with good accuracy. So thus AI helps optimize synthesis parameters for enhanced photocatalytic activity. So this is one method for photocatalytic, or the role of AI in the photocatalytic efficiency. Another one is predicting the adsorption capacity, how much the pollutants are adsorbed on the surface of a nanomaterial using the AI method. So AI models, again, it is used to here, and this particular process, it is used to estimate the adsorption capacity of nanomaterials for removing different pollutants, helping to predict performance before experimentations. So the key input parameters include like material properties such as surface area and its pore size, along with environmental factors such as pH, temperature, and initial pollutant concentrations. So regression models can be used to like quantitatively predict the adsorption capacity, which provides a numerical outputs for performance evaluation. And random forest and XGBoost, again, these models improve the prediction accuracy, and it can help to identify the influential factors affecting adsorption, such as surface area or pH. So ANN, in this particular case, can be used for the adsorption behavior, or the relationship between, to analyze the relationship between the multiple input variables as well as the adsorption behavior, so making it like highly suitable for real world systems. And support vector machines, SVM, is applied for both prediction and classification, which help to determine a nanomaterial is which nanomaterial, or whether the nanomaterial which we have created or which we have produced is suitable for a specific pollutant. Now, this is process optimization. That is, suppose we are fixing three parameters together at a time. We need to optimize the, suppose you are taking time, adsorption, dosage, or pH as three independent parameters. We need to optimize at which pH, at which time, or at which or the adsorbent dosage is used, or which is having good efficacy to adsorb the materials or adsorb the pollutants. So AI in this particular process is used to optimize the operational parameters in water treatment system to achieve maximum pollutant removal efficiency. Suppose you're taking pH, like temperature, contact time, catalyst dosage, initial pollutant concentration. So these are analyzed to determine the best operating conditions. So machine learning models, that is random forest, SVM, so these combination, or these large data sets are used to identify the optimal parameter combinations and highlight the most influential factors affecting the treatment efficiency. So ANN again, it captures the complex interaction between the multiple process variables, making it highly effective for modeling the real world system. And genetic algorithm is an optimization technique inspired by the natural evolution. So it searches for the best set of conditions by selecting and combining and refining parameters to achieve the maximum efficiency. So overall, like AI driven optimization reduces the resource consumption, it can save time, and it can lowers the operational cost. It also can improve the efficiency as well as the scalability of water treatment process, like making them more practical for the real world application. So additionally, AI enables the real-time monitoring and adaptive control also. Another one is AI-based water quality monitoring. So whatever we have done the process, finally we need to do the how quality, or when we need to check the quality of the water after the water treatment. For that also we can incorporate AI-based softwares also. So in this one, it will monitor the water quality, which allows continuous assessment of water safety and contamination level. So important parameters also we used as pH, turbidity, dissolved oxygen, heavy metals, and other contaminants are continuously tracked to evaluate the water quality. So in this machine learning models, so this will analyze the collected data to detect the patterns and predict contamination levels, which help in proactive decision making. And IoT and AI integration. So this is like a basic sensors which collect the real-time data and is processed by AI systems for continuous and automated monitoring without like manual interventions. And anomaly detection is so these algorithm will identify sudden changes or unusual patterns in water quality, enabling like quick detection of pollution events. So deep learning models can also used to improve the accuracy when dealing with the complex and large data set, enhancing the prediction and monitoring of the performances. So AI-based systems support early warning mechanism. It helps to prevent the health hazards and the environmental damage. So this is a model development, that is the culmination of how the process is going to be done. First, we will collect the data and experimental data, which we that is basically a literature review, and we will do the absorption, lab generated absorption and degradation data, and water quality data set. So these are the initial process we have to do. Next is we need to see the material properties such as size. So this is the mainly the characterization part. Material properties, size, morphology, surface charge, and pollutant properties. What is the molecular weight, solubility, environmental conditions like pH, temperature, etc. So this part is characterization part. Another next process is the model development. How you design the whole process as an experimental. So one, we need to choose the regression models, that is to predict the absorption capacity, classification model, that is suitable nanomaterial selection, and deep learning CNN to analyze this SEM-TEM images, that's the characterization images which we have already got, and then apply the AI-based water treatment models for the whatever the targeted process are. So this is my input for the AI driven nanomaterial base for water treatment. And thank you for listening. Thank you all of you.
Thank you, Dr. Jyotsna. And thank you, Poshali, for your question. But we will take the question for Dr. Jyotsna either tomorrow or later in the evening. We now move into the next segment, focusing on AI, brain systems, and computational models. Our next speaker is Sohit Reddy. He will be presenting his work on closed loop brain AI models.
And today I want to challenge how we think about performance, not just in AI, but in human AI systems overall. For years, we have optimized for output, like go faster, work harder, extract maximum productivity. But our brains don't work that way in real life. Our brains are regulatory systems. They're constantly balancing effort against fatigue, reward against risk. And that is the insight that changes everything. Today, I'm introducing closed loop brain AI models, systems that sense your cognitive state in real time and adapt to keep you performing sustainably, reliably, safely, and effectively. By the end, you will understand why this is not just nice to have, but essential for future of human AI collaboration. So let us start with a fundamental misconception. We treat performance like we're squeezing an orange. The harder you squeeze, the more juice comes out. But humans aren't oranges, right? Your brain doesn't operate at maximum effort continuously. In fact, it can't. Think about what happens when you exercise. A marathoner doesn't sprint the whole race, their body has a built in governor. The central governor model deployed by exercise scientist Tim Noakes shows that your brain constantly monitors fatigue signals, oxygen availability, and metabolic resources. So it basically paces you subconsciously to prevent collapse. And your brain is asking here, how do I maintain performance without breaking down? And the same applies to cognitive work. When you're deep in problem solving, your brain is balancing effort allocation, reward expectancy. Like it asks, is it worth it? And the risk. So if you push past that balance, you hit what is called burnout. So here is the reframe. Performance is not about maximal effort, it's about dynamic regulation. And if we can sense and support that regulation with AI, we can unlock sustainable high performance. And that's what closed loop means. So, now what closed loop is? It might sound technical, but it is actually how your brain already works. Let me break it down into five simple steps. Think of this as a blueprint for adaptive systems. First, continuous sensing. Your anterior cingulate cortex, otherwise called ACC, is monitoring your cognitive state right now, like your arousal, your error signals, like how much metabolic fuel you have left. It's like a biological dashboard. Second, feedback comparison. Your brain compares that current state to an optimal operating range. Like, am I in my zone or am I drifting into fatigue? And the third, adaptive adjustment. Based on that comparison, your brain modulates your behavior. You slow down, you ask for help, you switch tasks. Fourth, neural integration. This is not centralized. Your brain distributes this monitoring across networks like effort monitoring, fatigue tracking, resource availability. It's all well orchestrated. And last but not the least, fifth, equilibrium maintenance. The goal is not peak output right now, as I've discussed already. It's about stability and recovery dynamics over time. So, when we build such closed loop AI systems, we are mirroring this five step blueprint architecture. The system senses, compares, adjusts, and maintains equilibrium. So, here the neuroscience is pretty much clear. Your anterior cingulate and prefrontal cortex do this. We're just making it external, measurable, and actionable. And here is the uncomfortable truth about most AI systems today, they are open loop. A productivity app tells you to work harder, a learning platform increases difficulty when you succeed. None of the systems ask, how are you actually doing? Open loop systems optimize output without sensing your current state. If you're tired, they don't know. If you're making errors because You're cognitively overloaded, they push even harder. They are reactive. They correct errors after they happen. And the risk here is silent overload. You burn out before anyone notices. And that's where closed loop system changes this very fundamentally. The system senses your inferred brain state either through reaction time patterns or through error rates or through voice biomarkers or engagement patterns out there. It does not require brain implants. It infers from your behavior. Then it is proactive. Before you hit the wall, the system backs off, offers a break, or reframes the task. It maintains you within safe bounds. And for investors and policymakers out there, here is why it matters, because this is a shift from task completion focus to operator well being focus. It is not about extracting every ounce of productivity, it's about sustainable performance and human flourishing. And that's not just ethically right, it's economically smarter. Burnt out people don't create value. So let's get into the neuroscience. Every cognitive task costs energy. Researchers like Sharna Jamada have quantified it in such a way that task driven cognitive costs about an additional 5% of your baseline neural metabolism. So 5% doesn't sound like much until you're running on fumes. Your brain has a metabolic budget, and that budget is pretty much limited. So, but here is what's interesting. Your brain does not just allocate effort randomly. There is a principle called expected value of control. Your brain calculates reward times efficacy. Is the payoff worth the metabolic cost? If not, you reduce the effort. This is why motivation matters. It determines how you allocate your limited cognitive resources. And now in athletes, serotonin 5-HT adjusts pacing strategy during fatigue. It's basically preventing central nervous system breakdown. And the signal goes, we need to slow down or we'll break. In humans doing cognitive work, the principle is pretty much the same. Your brain is constantly adjusting task intensity to prevent overload. A closed loop system here intervenes before fatigue manifests as errors. So instead of waiting for mistakes, it senses the metabolic strain and lightens the load, and that is the real time modulation. And for students, researchers, and professionals out there, that's everything. And before we go further, I need to clarify something because many systems claim to be adaptive when they're really just personalized. Personalization is useful. It adjusts to your stable preferences, like you like dark mode, you prefer email over Slack, and it updates occasionally. Personalization works fine for study uh, state tasks. Closed loop adaptation is different. It responds to minute by minute fluctuations in your cognitive state. It updates continuously. It's essential for long variable tasks such as research, problem solving, cognitive work, creative work, like anything complex. And the recent work by Ahmed and colleagues uh, found out that adaptive AI system reduces cognitive load and significantly increases academic persistence in students with a correlation of 0.68 with cognitive load highly significant. Why? Because the system is meeting you where you are moment to moment, not where you were an hour ago. And most systems optimize for peak performance. Closed loop systems optimize for stability, and that's a radical shift. Consistency beats maximum output. Why? Because cognitive systems are pretty much fragile, and when you push to the limit, you don't gradually degrade, you cascade. One bad decision leads to another. Decision fatigue sets in, and your judgment collapses in the final. Research on decision fatigue by Baumeister shows that sequential decisions deplete your regulatory resources. But here is a fix: spacing and support mitigate that depletion. A closed loop system spaces decisions, offers support, and prevents cascading failure. And there is another piece, which is recovery dynamics. In athletes, exercise induced central fatigue precedes physical decline by minutes. Your brain gives you a warning signal before your body completely fails. A closed loop system basically monitors time to recovery and adjusts pacing proactively. And for medical professionals in high stakes scenarios, for pilots and for researchers under pressure, stability prevents catastrophic failures, not just in failures in output, but failures in judgment, safety, and well being. So, how do we sense cognitive state? We don't need brain implants, we can infer strain from behavior and often earlier than you're, you're consciously aware. Reaction time variably is a sensitive marker. When you're cognitively fatigued, your reaction time doesn't just slow, it becomes inconsistent. Your responses wobble, and this is pretty much detectable before you feel tired. So, when you're cognitively depleted, you hesitate before decisions and make omission errors, and you tend to skip things. And these patterns reveal something called resource depletion and EEG based metrics, which is theta to beta ratio. It's an entropy that measures cognitive load in real time, and this is an established neuroscience. And here's the elegant part, we can do this non invasively and multimodally. And voice biomarkers, your speech patterns change under cognitive load, and altogether, these enable adaptation without disruption. You're not wearing sensors, the system is inferring from what you're already doing. And for privacy conscious audiences out there and policymakers, this approach respects cognitive boundaries. No invasive neural monitoring, it's just behavioral inference out there. And technology is never neutral, so let me be explicit about the ethics here. First, support, not override. Here, the closed loop systems scaffold your decision making. They don't make decisions for you, you're still in control. Here, the system is like a coach, offering guidance and not taking over. And the second, transparency. Users must understand why and when the system adapts. If the system reduces task difficulty, you should know why. And this maintains trust and agency. And third, think of cognitive safety like a physiological homeostasis. Your body maintains temperature, pH, oxygen levels without you thinking about it. So, cognitive regulation does the same for your executive function, it's protective. Fourth, opt in, not imposed. Individuals retain control over which adaptations they accept. You can say, actually, I want to push hard right now, something like that. And fifth, privacy in measurement. Behavioral inference respects cognitive boundaries. You're not being psychologically profiled. The system is inferring task relevant cognitive load, not surveilling your thoughts. For investors, this is also a business advantage, I would say, because trust and transparency create user loyalty and regulatory resilience. And let's talk about burnout. We treat it as a personal problem, often a sign of weak resilience or poor self care. And that's wrong. Burnout is a systems failure, it's not individual's weakness. So it's design alignment between task demands and it's basically a design misalignment between task demands and human capacity, I would say. You put someone in a system that demands constant high performance without recovery, and burnout is pretty much inevitable. So basically, here the closed loop system prevents this by matching overload to capacity in real time. So this has massive implications for institutions such as universities, research labs, hospitals, tech companies. So if your system is built right, burnout prevention is automatic. And for policymakers out there, this is about institutional design. And for students and researchers out there, this is about finally having systems that work with you instead of against you. And here is the fundamental shift driving all of this. Machines adapt to brains and not vice versa. For decades, we have been trying to change humans to fit AI, work like a machine, think like an algorithm, operate 24 by 7. It's backwards. So closed loop brain AI systems flip this. Biological constraints become design constraints. Your brain has a neural cost budget, fatigue dynamics are real. Your recovery is non-negotiable. And these aren't just limitations to overcome, they are requirements that shape system architecture. And we can honor biological rhythms. Your brain has ultradian cycles, roughly 90 to 120 minutes of focused attention, and then natural decline. And then we have circadian rhythms. So a human compatible system respect these and not fight them. So, and there is a research imperative here. Closed loop AI systems are foundation for the next generation of human AI collaboration. And this is not optional, this is the direction the field must move. So where does this leave us? We are moving from we are moving from optimization of output to regulation of equilibrium. And that's a complete reframing of what performance means. Not how much can I extract, but how do I maintain sustainable excellence? Not how hard can I push, but how do I stay healthy while performing? Here, the closed loop brain AI systems represent the next frontier in human compatible intelligence design. And for scientists, this is a rich research space. Like inference algorithms, you have neural monitoring, you have ethical frameworks. And for investors, this is a huge market opportunity. People will pay for systems that work with their biology and not against it. And for policymakers out there, this is a mandate because the institutions must be designed around human well being. And for every student and researcher and professionals out there, this is about having tools that finally serve you and not exploit you. Thank you.
Thank you, Sohit. If any one of you have questions for Sohit, you can post it in the chat. We do have two questions from the YouTube live. Sohit, are you ready?
Yeah, yes, I'm ready.
Okay. So the first question for you is, how do reviews distinguish open loop versus closed loop intelligence in brain AI?
Yeah, that's a great question. So basically, reviews here, what I've seen, they define open loop as like one shot and feed forward predictions without feedback. For example, static EEG classifiers, like which are reliable for simple tasks, but they're little brittle to noise, I would say. Whereas this closed loop intelligence adds real time monitoring and correction, like ERRP detection, refining, and boosting accuracy by 20 to 40% via adaptation. So this mirrors control theory, which I've mentioned already, which is like open for speed and closed for robustness. Yeah, that's all I would say.
Okay, I hope that answers. And the second question for you, Sohit, is that how might hybrids propel brain AI toward truly intelligent systems?
It was hybrids, right?
Yes.
Yeah, so hybrids basically like fuse personalized cores with adaptive loops for living models, like, the resilience, bias resistance, and predictive in nature. So literature envisions this thing called enterprise grade neuro AI. They're potentially accelerating by quantum sampling, marking the shift from reactive to proactive intelligence. So this could be one example of a hybrid system. Yeah, thank you.
Okay, thank you, Sohit. Is there any more questions? I can give it a few seconds. Any one of you can even raise your hands if you want me to wait for some time until you type your question. Okay, I think we can proceed further. Up next is Prajwal Anand. He has completed his BE in Mechanical Engineering from AMC Engineering College, and now he will be presenting on AI-based detection of brain tumors. Welcome, Prajwal.
Hello everyone. Today we are going to talk about AI-driven subtype discovery and evolution modeling in glioblastoma. Glioblastoma remains as one of the deadliest tumors at the moment, without any proper cure. And the goal of this research is basically precision treatment, where we design therapies around subtype vulnerabilities and resistance patterns. Here, the focus is mainly on hidden subtype discovery by using multimodal data to reveal biologically distinct patient groups, and then to track the disease as a changing trajectory rather than a single snapshot. And the survival rate of glioblastoma remains 12 to 15 months mostly common, and then 18 plus months more off on the rare end, but yes, upper rough benchmark of that. And also we have observed that patients behave differently, but the treatment is still uniform, even though patients have been surviving for some for 6 months and some even 30 plus months. The spread shows that it is diverse, and the treatment has to be also in a certain way. And glioblastoma behaves like a changing ecosystem. So with a disease like glioblastoma, where it also behaves like a changing ecosystem, uniform treatment is not the optimal path of it. And the trial and error method which has been going on also doesn't help. So with the help of AI, we are trying to track those, because AI comes here where it can interpret signals, all the signals into one. And it might be imaging, it might be genomics, clinical data, and time progression. It can interpret the signals together and give precision understanding and find out hidden subgroups that are not visible from normal reports without combining all the reports together. So the pipeline of this is simple: to collect all the patient data, identify hidden subgroups and cluster those into subtypes, and then model the evolution of tumor across all those subtypes and simulate treatments across it. So it requires MRI scans, genomic profiles, records, treatment history, survival data, and public data sets. And clustering all of them will reveal the hidden states, and also we can define new subtypes that are clinically and biologically interpretable. And it will be modeled as a trajectory as mentioned earlier. So since tumors also evolve under selective pressure from treatment and microenvironment, this can estimate the likely progression paths across time and target the growth, mutation, and resistance of it. And also in terms of test alternative treatment futures before real world use, what we are trying to do is run multiple plausible disease paths to predict similar paths that it can take, and then design treatments particularly to that, and also to compare the relative benefit before clinical use, and to avoid purely trial and error treatment choices. That will also be one of the main things we are trying to target, which is treatment and then report, and then to treatment again based on the report outcomes. So it can be achieved through creating a live virtual living virtual patient model, a digital twin representing the patient's tumor and biological state. That is the idea of it, that whenever the new data becomes available, it updates that itself. And also we can track the evolution paths with the help of digital replica, which I have mentioned earlier. So that can be done through here. And each subtype may expose different therapeutic vulnerabilities, because each one of them will have different vulnerabilities, and sometimes one vulnerability of the one thing might not be the vulnerability of others, and that might affect in a different way than expected. So this will help us in finding the combinations of this, on which will be the optimal path to target those subgroups without affecting the other ones, and then the treatment can be designed in such way. And this is the benefits and expected impact of it is personalized treatment and better survival prediction, and also faster therapy discovery rather than trial and error. It will be able to discover the path of therapy faster before the expensive testing starts, and less trial and error. So the long-term vision of this is basically to map the space, to build a comprehensive atlas of glioblastoma progression states, and predict the estimate disease trajectories that it can appear and take next, and then to control the progression, which with the help of treatments which is being done right now. And that is the reason patients are surviving longer, with the help of controlling the progression where it is stopped at the stage it is right now. And but still there are some challenges remain to achieve this, which is in terms of data availability and biological complexity, that it can evolve faster than how the model is predicting in terms of accuracy and then clinical validation, because the framework must prove value in prospective patient care. And the main insight from where it all started is that long-term survivors are not lucky. They represent different biological systems, because something has to be there which we are not found yet, based on which the survivors are so diverse and vast. So that is the main insight which all of this research started from. And to treat glioblastoma as not one disease, and AI will enable the discovery of subtitles and predict evolution resistance rather than the
Okay, give it a minute. I think there's a technical glitch or something.
treatment can be designed in such way. And this is the progression states and predict the estimate disease trajectories that it can appear and take next, and then to control the progression, which with the help of treatments which is being done right now. And that is the reason patients are surviving longer with the help of controlling the progression where it is stopped at the stage it is right now. And but still there are some challenges remain to achieve this, which is in terms of data availability and biological complexity, that it can evolve faster than how the model is predicting in terms of accuracy and then clinical validation, because the framework must prove value in prospective patient care. And the main insight from where it all started is that long-term survivors are not lucky. They represent different biological systems, because something has to be there which we are not found yet, based on which the survivors are so diverse and vast. So that is the main insight which all of this research started from. And to treat glioblastoma as not one disease, and AI will enable the discovery of subtitles and predict evolution resistance rather than the
Okay. Thank you, Prajwal. Prajwal, you are present here, right?
Yes, yeah, I'm present here.
Okay. So there are some questions. I think other than just Sohit, if anyone has any question for Prajwal, please type it out. Prajwal, the first question from Sohit for you is, you have mentioned about mapping tumor evolution paths. How is mapping of tumor across different states in different pathways done? Isn't data limited yet complex to deal with? What do you think of AI virtual cell?
Yes, so in terms of mapping the evolution path, right now, what is happening is many research are going on currently regarding for this particular tumor, where every day they're observing, every day the tests are being done. And those data, initially, if they are not publicly available also, they are there in terms of for research and people who are doing the research within the clinics also, they are particularly going through it. So in terms of using that with AI, in terms of virtual stuff, and also in terms of the way I mentioned in terms of creating the 12 of it. So every day this data is there, and also all the causes in terms of it, basically a reaction, right? Reaction to the medicines that can be tracked. And that goes into a very complex level. Yes, it is a very complex thing to achieve. But many similar things have been done in other fields. Even the new meta model, which tracks the brain activity for watching. So it can be similar way. There are many other research also done where similar things of brain activities have been tracked. So I was thinking in that similar way we can do this also and track it based on if it is mapping. Let's say we can take a previous day record and predict the next what can happen within the for the particular treatment, and then compare it with the actual thing that happened outcome. So that is the way we can build it in step by step, but yeah, it will be a pretty complex to map it across. and public data is very limited for this.
Okay, I hope that answers your question, Sohit.
Yes.
Okay, so now we move to Hari Sudarshan. He has completed his master's in robotics and AI from the University of Glasgow, and his presentation focuses on agentic AI.
Hello everyone, my name is Hari Sudarshan and I want to talk about something that I think is one of the most significant shift happening in AI right now. Not smarter models, not better chatbots, but a fundamentally different way of building AI systems. We have spent years making AI better at answering questions. What's changing now is making better at actually doing things. So, why is everyone suddenly talking about agentic AI? The concept isn't new. Researchers have been thinking about autonomous AI systems for decades. What changed is that three things finally came together at the same time. Model becomes reliable enough to orchestrate tools, not just generate text. Inference cost dropped dramatically. So, running a multi-step loop that calls an LLM 10 times in a row is now economically feasible. And the ecosystem is matured as well for APIs, vector database, code execution environments. Everything an agent needs to act in the world is now plug and play. The idea isn't new, the capability finally is. So, what actually makes something an agent rather than a chatbot? It comes down to three components working together. You have the LLM as the reasoning layer. It decides what to do next. You have tools such as APIs, search engines, code runners that let it reach into the real world. And you have a workflow loop that keeps running until the task complete. Not until the text generated, but until the task complete. One design insight worth calling out here. The best platform performing agent system separates running from execution. When the same model decides what to do and immediately does it in one shot, the error cascade. When you split those two responsibilities, reliability improves significantly. Let me make this concrete. The loop looks like this. The agent receives a goal. It thinks, what do I actually need to accomplish this? It acts, it calls a tool, maybe a API search or a database query. It observes the result, reads what came back. Then it asks, am I done? If not, it loops back and plans the next step. This is fundamentally different from a chatbot, which always exists after one response, regardless of whether the task is complete. An agent only exists when the goal is met or when it gets stuck, which is actually one of the key challenges we will get into. So, here is a real example that makes this tangible. Imagine a user types analyze Apple's performance this quarter. A chatbot reads that and generate a response based on whatever it already knows and then stops. An agent does something entirely different. It first interprets the intent, like what scope, what time frame, and what metrics matters. Then it fetches live market data through API. Then it pulls relevant fillings or news. Then it analyzes the patterns across all that. Then later it generates a structured report. Five distinct steps, all triggered by one sentence. No human in loop interaction between them. I built early versions of this kind of pipeline in my own projects, and the potential is immediately visible. What equally visible is how quickly things breaks when one step in that chain produces a bad result. Which brings me to the part where most people skip the problems. Hallucination in a chatbot means you get wrong answer, but hallucination in an agent means the system acts on wrong answer and then takes three more steps based on it before anyone notices. That's actually a qualitatively different kind of failure. Error propagation is real because one bad tool call can silently corrupt everything downstream. Tool misuse is subtle. The agent might call the right API with wrong parameters and get back data that looks plausible but isn't. State tracking degrades over long tasks as context fills up and cost scales fast. Every loop iteration is an LLM call. So my experience building these systems taught me one thing clearly. Reliability is the bottleneck, not the intelligence. Despite those challenges, the application potential is significant and specific. In enterprise settings, agents can replace brittle rule-based script with systems that reason about exceptions. In decision support, they can research, summarize, and surface insights that a human analyst would take hours to compile. In workflow optimization, they can monitor pipelines, trigger actions, and escalate issues with constant babysitting. Looking ahead, multi-agent systems where specialized agents collaborate, persistent memory across sessions, and the human-in-loop systems designs for high-stake decisions. These all are the active areas of development. The bottleneck holding all this is not intelligence, it's the system design, reliability, and control. So let me leave you with three things. AI is moving from answers to actions. That shift is already underway. Agents are not smarter chatbots, they are different architecture with different failure modes and different design requirements. And the challenge now is not capability, it's the control. I will close with a question that I think worth sitting with. If an AI agent makes some consequential mistake mid-task, not just giving a wrong answer, but takes a wrong action, then who is responsible? That question doesn't have a clean answer yet, and I think how we answer it will shape how this technology develops. So, thank you so much, everyone, for listening. Thank you once again.
Thank you, Hari. Next, we have Shubh Garg, who is currently pursuing his degree in electronics and computer engineering at Thapar Institute. He will be speaking about LLM versus non-LLM approaches in healthcare.
Okay, so today I'm going to talk about one more specific intersection, that is large language models and mental health, drawn from a landscape of study of nearly 20,000 healthcare NLP papers published this year in NPJ Healthcare Systems. Okay, so moving on. Now we're talking about the scale of the problem. So as we know, there are 970 million people globally live with mental health condition, out of which 75% in middle and low income countries receive no treatment, and there has been a psychiatric to patient ratio of approximately 1 to 14,000, which which is the gap that we're trying to address. Okay, so moving on. Okay, so the study that we have conducted is this, in which we have retrieved papers of 44,000 papers approximately, and we have analyzed narrowed down to 19,000 papers, in which we have classified into two categories, which is LLM versus non-LLMs, and then we have narrowed it down to 28 research teams. This is a data-driven map where healthcare NLP actually stands in. Okay, so the key finding that we have done is the mental health is the second largest LLM application cluster in healthcare at 11.62% of all LLM studies. That's not a coincidence. It reflects something structural why LLM belongs in the space. Okay, so moving on to next, why LLM fit mental health tasks. So mental health task properties are open-ended, unstructured language, emotionally led, and long multi-turn conversations, cross-domain contextual reasoning, non-standard. Whereas LLM comes into place is a pre-trained on vast natural language data, semantic understanding and effect of nuances, multi-turn dialogue management, and future generalization across topics, robust informal and idiomatic languages. So coming on to the applicative side, so there are three active areas that can supportive. So first is the supportive dialogue agents like VBot for real-time support, clinical simulation for training psychiatry students safely, and then we could have population screening in which we can talk about the free clinical notes for early detection of depression and PTSDs. Okay, as we know, for everything there's a two sides of the coin. So now talking about the flip side of the coin, where we talk about the risk and the limitations. So first is hallucination in high stake context. Second, we could have the lack of explainability. Third, we have bias in training corpora. Fourth is privacy and data governance, in which the serious data governance concerns could be a deployment without guardrails is a premature thing. Okay, so as we know, we have the problems which are very much solvable. So we can come at a middle path and solve one problem which is called as a hybrid pipeline. So as we know, non-LLMs are good at EHR decoding of 21.6% in non-LLM studies. Then we could have used NER of 12.3%. Then ontology mapping of 7.7%. Then we could complement it with LLM excels in generation, interaction and conversation, and multimodal perception. So we could make a structured pipeline in which we could make a hybrid out of it, in which the input could be the patient input, patient text or patient language. Then we could process it with a black box LLM layer. Then we could move it to structured NLP, focus on the clinical output. Okay, so as we know, there has been a massive growth in 1 year, which is 3.7x of 2023. So like there was 757 in 2023 and 2787 papers in 2024, with GPT-5 coming in and Gemini 2.5 and DeepSeek R1 entering clinical reasoning. Mental health will be one of the fastest moving application areas in the next 2 years. So as we know, LLM won't replace therapists. The goal is extension, reaching the 75% who currently get nothing. The field is moving whether we are ready or not. The question is whether we build responsibly or not. Thank you.
Thank you, Shubh. Next, we have K. Vijayalakshmi. Her topic is predictive analytics for fitness and workload optimization.
My name is Vijayalakshmi. I have completed my MTech in VLSI. Today I will be presenting a study titled Keeping Athletes Match Fit All Season. Predictive Analytics for Workload Optimization using Machine Learning. This work explores how data-driven intelligence can help prevent overtraining and injuries proactively, rather than responding after performance breakdowns occur. Elite athletes are exposed to high physical, mental, and competitive demands throughout long seasons. While training intensity is necessary for peak performance, excessive or poorly managed workload leads to fatigue, injuries, and performance decline. Traditional training programs often rely on experience based judgment, fixed workload plans, reactive injury management. This research proposes a new predictive analytics approach where machine learning models analyze athlete data continuously to optimize workload before overtraining occurs. The central idea is to balance performance enhancement with injury prevention using intelligent systems. The challenge of workload management is complex because human psychology is not uniform. Key challenges includes athletes responding differently to identical training loads, fatigue accumulating invisibly before symptoms appear, manual monitoring failing to capture subtle risk signals, delayed intervention windows once injury occurs. Most current systems react after muscle soreness, fatigue, or injury symptoms appear, which significantly reduces recovery efficiency. Hence, there is a growing demand for early personalized and data driven workload prediction systems. Machine learning enables systems to learn patterns from historical athlete data and predict future outcomes. In this framework, supervised learning maps training load to injury or fatigue outcomes. Feature engineering identifies critical predictors such as intensity, recovery, and training frequency. Time series modeling captures workload, accumulation, and fatigue trends over time. Classification models categorize athletes into different risk levels. Unlike rule based systems, ML models adapt and improve continuously as new data becomes available. A reliable predictive system depends on a robust data structure. The pipeline includes data collection layer, variables, GPS trackers, wellness questionnaires, strength tests, integration layer, standardizing data from multiple sources, processing layer, real time analytics and risk prediction, visualization layer, dashboards for coaches, psychotherapists, and analysts. This architecture ensures continuous monitoring, early warning detection, and informed decision making. The model incorporates multiple dimensions of athlete health: external load, distance, speed, acceleration, training duration, internal load, heart rate, variability, perceived exertion, recovery indicators, sleep quality, muscle soreness, fatigue levels, contextual factors, match density, travel fatigue, environmental conditions. By combining psychological, behavioral, and contextual data, the system achieves more accurate and individualized risk predictions. Model training follows a structured methodology. Minimum 6 to 12 months of historical data ensures reliability. Clear labeling of overtraining and injury events. Feature selection reduces noise and improves performance. Cross validation tests generalization on unseen data. The focus is on building stable, interpretable, and reliable models that coaches can trust in real world environments. Predictions are converted into actionable risk scores. Low risk, where green indicates athlete training is optimal. Yellow indicates moderate risk, early fatigue indicators detected, whereas red indicates high risk, high probability of overtraining or injury. Thresholds are adaptive, adjusting based on athlete fitness progression. This framework allows timely inversion without unnecessary training disruption. Implementation considerations. Deploying predictive models in practice involves challenges such as ensuring model interpretability for coaching staff, preventing alert fatigue through smart threshold design, maintaining data privacy and athlete trust, continuously updating models as athlete conditions change. Successful implementation requires alignment between technology, coaching, philosophy, and athlete well being. For maximum effectiveness, analytics must integrate into daily routines. Morning wellness assessments update risk scores. Weekly planning meetings use predictions to adjust low. Pre competition analysis identifies high risk athletes. Feedback loops refine models using real outcomes. This creates a closed loop decision systems, improving both performance and safety. For future evaluation, future advancements include multimodal learning, integrating physical, psychological, and biomedical data. Explainable AI for transparent decision support. Edge computing for real-time on-field predictions. Fully personalized athletes specific digital twins. These developments will transform workload management into a precision performance science. In conclusion, predictive analytics enables a shift from reactive injury management to proactive performance optimization. By intelligently managing workloads, teams can reduce injury incidents, enhance consistency, extend athletic careers. This approach represents a significant advancement in data driven sports performance management. Thank you.
Thank you, Vijay ji. We now have Porigola Shruti. She comes from a background in geology. Her presentation will be on AI driven sustainable rare earth exploration.
Good morning everyone, and myself Shruti. Today I am going to present my topic on environmentally and health conscious rare earth exploration using AI and multi-sensor geophysics to reduce ecological and human risks. In this presentation, I will cover the introduction, problems in rare earth exploration, environmental and health issues, how AI is used, and the workflow and the role of AI changes we can make, and the limitation, and finally the conclusion. Rare earth elements are very important materials used in modern technologies like artificial intelligence, renewable energy, and electronics. Because of because of increasing technology, the demand for these elements is growing very fast. But the problem is the way we explore and extract these elements causes serious environmental damage and health risks. Traditional methods are not accurate and depend on trial and error, so there is a need for better methods. That is where AI and geophysical techniques help by making exploration more accurate and reducing damage. The main problem in rare earth exploration is uncertainty. We don't know how exactly where the minerals are, so large areas are disturbed. This leads to unnecessary land clearing and damage. Also, many drilling attempts are done, but not all are successful. This increases environmental damage and waste resources. At the same time, workers and nearby people are exposed to harmful dust and radiation. Environmental issues. Exploration activities causes several environmental problems. First, deforestation happens because trees are cleared, soil quality reduces due to continuous disturbance, water get contaminated due to chemicals and metals, air pollution increases because of dust. All these together damage the ecosystem and affect biodiversity. These activities also affect human health. Dust can cause respiratory diseases. Exposure to radioactive elements like thorium and uranium can lead to serious health risks. Contaminated water can cause organ damage. People living near these areas are also affected by pollution, which increases health problems. AI does not directly find minerals, but it helps reduce uncertainty. It uses data from different techniques like satellite imaging and geophysical surveys. These include hyperspectral imaging, magnetometry, resistivity, and radiometric surveys. AI analyzes all these data together to understand patterns and improve accuracy. Next. This is the workflow. First, we collect data from satellites and sensors. Then, we combine all the data. AI analyzes this data and finds patterns. Based on this, it predicts where minerals are likely present. Then, we do targeted exploration instead of exploring everywhere. This reduces environmental and health impact. Role of AI. AI plays an important role here. It combines different data sources into one system. It can process large data quickly. It detects patterns that humans cannot easily see. It predicts mineral rich areas. Most importantly, it reduces uncertainty and improves decision making. Changes we can make using this approach, we can make several improvements. For the environment, we can reduce land disturbance, avoid sensitive areas, reduce waste, and protect water resources. For health, we can reduce worker pressure, avoid hazardous zones, improve safety, and protect nearby communities. So overall, this approach makes exploration safer and more sustainable. The limitation. However, there are some limitations. AI need high quality data, otherwise result may be wrong. This technology is expensive. It is not available everywhere. Skilled people are required to use it. Also, this method does not completely stop mining, it only reduces the impact. Conclusion. In conclusion, rare earth exploration is necessary but harmful. If done traditionally, AI and geophysical techniques help to make exploration more accurate. This reduces environmental damage and health risks, but mining cannot be completely avoided. So, goal is to make it safer and more sustainable. And thank you for giving me this opportunity to explain about my knowledge on the rare earth elements.
Thank you, Shruti. Next, we have Jitnyasa Zade, a BTech student interested in AI and data science. She will be presenting her work on DiaRisk AI Assistant.
Hello everyone, I'm Jidnasa Zade, and today I'm presenting DiaRisk, an AI driven initiative aimed at transforming how we approach personal health monitoring. So, in an era where data is everywhere, I just wanted to create a tool that turns complex medical metrics into actionable life saving insights. But why is the tool like this necessary right now? Let's look at the current landscape. So, we are currently facing a global health crisis, not just from any acute illness, but from chronic lifestyle diseases like diabetes, blood pressure, etc., that often go unnoticed. So, the problem is twofold: lack of early stage awareness and high barrier to professional diagnostic services. And many people don't realize they are at risk until it's too late. So, DIRISC was born from the need to provide a preliminary digital checkup that anyone can access from their home. Uh, to address this gap, I defined three core pillars for this project. My objective for this project was very clear, like first to develop a high accuracy predictive engine for diabetes risk. Second, to build an interactive assistant capable of understanding natural language symptom. And third, to break down language barrier in healthcare by making the tool multilingual. So this isn't just a calculator, it's comprehensive health companion. So how does DIRISC actually function for the end user? So let's get into it. So DIRISC offers a multifaceted approach to health. It features a dedicated disease prediction module for clinical data, like a symptom checking chatbot for general inquiries and a personal health dashboard. So, by combining all of these, the user gets both a quantitative risk score and a qualitative conversation about their well being. So, one of the most important aspects of accessibility is speaking the user's language. Healthcare is universal, right? And language is often a barrier. So, to address this problem, I integrated the Google Translate API. This allows the system to support user in their native tongue, whether it is any regional language from India or any international language like Korean, German, Russian, etc. So this ensures that the AI's advice is understood perfectly, which is critical in a medical context. So the bridge between this complex AI and the user is a clean, intuitive interface. So for the user interface, I utilize Streamlit. I chose this framework because it allows for seamless integration of Python-based data models into a clean web-based dashboard. And the design philosophy was simplicity first. So I wanted to ensure that even a non-tech user could input their vitals and receive a clear result in like within seconds. So now let's step behind the curtain and look at the technical architecture that makes this possible. Um, so under the hood, DIRISC often follows a modular architecture. The Streamlit frontend captures user inputs, which are then passed to a fast API backend. This backend acts as a brain, routing data either to the machine learning models for prediction or to the language natural processing, which is also known as NLP. So, engine for the chatbot conversations. So, the efficiency of the system depends entirely on the tools we use, and tech stack is built entirely on Python, leveraging its robust data science ecosystem. I use scikit-learn for the predictive modeling and transformers. So specifically, I implemented Distill GPT as well to ensure chatbot provides quick responses without requiring massive computational resources, and Pandas and NumPy for handling like data preprocessing. Of course, an AI is only as good as the data it learns from. So the predictive power of DIRIS comes from TMA Indian's diabetes dataset. This dataset is gold standard in medical ML, containing critical features like BMI, age, thickness, skin thickness, glucose level, insulin, age, etc. So these 8 variables are the key indicators where we use to determine a user's diabetic risk profile. So before training the model, I had to understand how these variables interact. So before any training happened, I conducted a deep dive EDA, which is exploratory data analysis. So by generating heat maps and correlation matrices, I identified that glucose and BMI had the highest correlation with the diabetic outcomes. So this phase was very crucial feature for the feature engineering and ensure that the model wasn't just guessing but focusing on most statistically significant health markers. So So I experimented with several algorithms, including KNN, SVM, and etc. But from those, random forest classifier was the ultimate winner. So, this balance of precision and recall is vital because in healthcare, minimizing false negatives is just as important as overall accuracy. So, here I will talk about real world challenges and ethics. So, working with medical data brings significant ethical responsibilities, and one major challenge is model interpretability. So, users need to know why the AI thinks that they are at risk, and furthermore, I have integrated privacy first protocols to ensure that personal health data is processed securely and is never stored without explicit intent. So we must always remember this tool is an assistant to not a replacement for a doctor. So the roadmap for DiRisk is ambitious. I'm looking into wearable integration to pull live data from Apple Health or Google Fit. I also plan to expand into medical imaging using CNN to analyze X-rays or skin scans. Finally, I want to add a geolocation feature that automatically finds the nearest specialist if the AI detects a high risk scenario. So, to wrap everything up, so in conclusion, DIRISC is about empowering individuals through early detection. By merging AI with accessibility, we can help people take control of their health before symptoms becomes a crisis. So, thank you everyone for your time and for joining me in exploring the future of personal health technology. Thank you.
Okay. Thank you, Shruti. Thank you, Poshali, for your question. We can take your question later after the conference and connect you with Jidnyasa. Now, we will be taking a very short break for 15 minutes. We will be back at 5:10 sharply and we will continue. Thank you. Welcome back, everyone. We now continue with AI in biology, medicine, and healthcare. Our first speaker of this segment is Shreya Das. She has completed a master's in biological sciences from Presidency University, Kolkata, and her presentation focuses on exercise induced myokine signaling and cancer prevention. Welcome, Shreya.
Hello everyone. Today I'll be discussing one of the most compelling intersections in preventive oncology, how physical activity is now recognized as one of the most powerful non-pharmacological strategies for cancer prevention. Beyond its effect on body weight and cardiovascular health, exercise triggers profound molecular changes throughout the body. During exercise, our skeletal muscles don't just generate mechanical force, they transform into active endocrine organs. As muscle fibers contract, they release hundreds of different proteins called myokines directly into circulation. These myokines then travel throughout the body, creating a systemic biochemical environment that influences cancer cell behavior, enhance immune surveillance, and reshape the tumor microenvironment. Understanding this muscle cancer communication reframes exercise from a lifestyle factor to a biological active intervention with therapeutic potential. To understand how exercise communicates systematically, first we need to understand what myokines are. Myokines are cytokines and peptide hormones that are synthesized and secreted specifically by skeletal muscle fibers in response to contraction. More than 600 myokines have been identified, each with distinct biological roles. Among the most well-characterized anticancer myokines are the interleukin 6, interleukin 15, irisin, spark, decorin. Importantly, myokine release is not uniform. The crucial point is that exercise type, intensity, and duration all matter. Resistance training, aerobic exercise, and high intensity interval training each produce distinct myokine signatures. This means exercise acts as a programmable biological stimulus, generating a dynamic signaling network affecting everything from metabolism to immune function to cancer risk. With that foundation, let's examine how myokines directly affect tumor cells themselves. Several myokines possess direct anti-tumor activity through multiple molecular mechanisms. Take interleukin-6 as a prime example. This is particularly interesting because IL-6 acts differently depending on context. As illustrated here, the chronic sustained elevation of IL-6 seen in obesity and inflammation actually promotes cancer, but the acute pulsatile release during exercise has the opposite effect, activating tumor suppressor pathways like p53 and inhibiting pro-growth signaling. Decorin, another key molecule, binds and neutralizes multiple receptor tyrosine kinases on cancer cell surfaces, effectively blocking proliferative signaling. Spark modifies the extracellular matrix architecture, making it mechanically and biochemically less permissive for tumor invasion and metastatic spread. Together, these effects mean that regular exercise establishes a biochemical environment that actively resists cancer development. Beyond directly affecting tumor cells, myokines powerfully enhance our body's anti-tumor immunity as well. Here, myokines play a central role in strengthening the immune surveillance. Interleukin 15 is particularly important as it drives the activation, proliferation, and cytotoxic functioning of the natural killer cells, which serve as the body's frontline defense, identifying and eliminating precancerous and transformed cells before they can establish tumors. Exercise also increases the immune cell trafficking, improving the ability of T cells and NK cells to reach the peripheral tissues where they can detect problems early. Myokines enhance T cell toxicity and improve tumor infiltration by cytotoxic lymphocytes, which is critical for eliminating the established cancerous cells. Importantly, these immune benefits follow a dose response pattern. Moderate regular exercise maximizes immune function, whereas exercise training without recovery can transiently suppress it, highlighting the importance of appropriate exercise prescription. In parallel with immune modulation, myokines profoundly shape the systemic metabolism as well. Here, exercise-induced myokines create profound metabolic shifts that reduce cancer risk across multiple pathways. Irisin is a relatively new molecule that promotes the browning of white adipose tissue, essentially converting the energy-storing fat into energy-burning fat, which increases metabolic rate and help combat obesity, itself a major cancer risk factor. As shown in this metabolic pathway figure, myokines enhance insulin sensitivity across all tissues, lowering circulating levels of insulin as well as insulin-like growth factor 1, both of which are potent cancer-promoting growth factors. There is even evidence that myokines shift cellular metabolism away from aerobic glycolysis, the so-called Warburg effect that cancer cells exploit, and towards oxidative phosphorylation, which is less efficient for rapid cancer cell division. Hence, this metabolic reprogramming creates a systemic environment that simply does not support cancer, even regulate. For any tumor to grow beyond a microscopic size, it must develop its own blood supply through angiogenesis. Myokines regulate this process in some sophisticated ways, favoring normal tissue while limiting the tumor vascularization. As depicted here, some myokines promote the formation of a normalized, well-structured blood vessels with non-leaky walls in the healthy tissues, while directly antagonizing pro-angiogenic factors like vascular endothelial growth factor. This dual regulation suppresses the chaotic, leaky vasculature typical of tumors, potentially starving the nascent tumors of the blood supply they need to progress, while preserving the healthy tissue perfusion and oxygen delivery. This represents a form of endogenous angiogenic therapy delivered through muscle contraction. Closely linked to angiogenesis is chronic inflammation, another key driver of cancer risk. Chronic low-grade systemic inflammation, often called inflammaging in older adults, is a well-established cancer risk factor. Although exercise induces acute inflammation, regular training produces a long-term anti-inflammatory state through myokine signaling. Exercise-induced myokines shift the systemic cytokine balance from pro-inflammatory mediators like TNF-alpha and chronic IL-6 towards anti-inflammatory profiles including IL-10. By reducing visceral adiposity, one of the body's largest sources of inflammatory cytokines, exercise removes a major contributor to this chronic inflammation. There's even emerging evidence that myokines improve the intestinal barrier integrity, reducing the translocation of bacterial products into circulation that can trigger inflammatory cascades. Hence, an anti-inflammatory environment is created, less permissive for malignant transformation. These effects are especially important in cancers driven by hormonal signaling. For cancers driven by sex hormones, particularly breast cancer in women and prostate cancer in men, myokines influence both hormone production as well as receptor signaling. Exercise-induced myokines reduce the circulating estrogen levels by decreasing the aromatase activity in adipose tissue, which is the primary site for estrogen production in postmenopausal women. They even modulate the sex hormone binding globulin levels, altering the hormone bioavailability. In prostate tissue, certain myokines can reduce androgen receptor sensitivity, potentially limiting the growth signals these receptors provide. These mechanisms as a whole help explain why exercise shows particularly strong protective effects in hormone-driven malignancies. These hormonal effects are particularly relevant for prevention in high-risk populations, especially women with BRCA mutations or even strong family histories. The relationship between cancer and exercise prevention follows what we call a dose response curve, though it's simply not linear. Epidemiological data show that approximately 150 minutes of moderate intensity exercise per week reduces cancer risk by 20 to 30% for several cancer types. As shown in this curve, higher exercise volumes can provide additional benefits, but the incremental gains diminish, and there's an important caveat. Exercise without adequate recovery can paradoxically increase oxidative stress and inflammation, potentially negating some benefits. The optimal prescription involves consistency over time, variety in exercise modalities combining aerobic, resistance, and high intensity work, and appropriate recovery periods. This ensures sustained beneficial myokine signaling without pushing into the counterproductive overtraining zone. Understanding these mechanisms open the door to translational and clinical applications. The mechanical insights we have discussed today open multiple promising intervention pathways. One approach is the pharmaceutical development, that is the formation of myokine mimetics or enhancers for population who cannot exercise due to disability or chronic illness. Though we should be clear that these would complement and not replace physical activity where possible. Another is precision exercise prescription, tailoring activity based on individual myokine responses and cancer risk. Third is the combination approaches, that is exercise combined with conventional therapies may even produce synergistic effects through shared signaling pathways. As we integrate this knowledge into clinical practice, several priorities even emerge. First is that the exercise prescription should be considered as rigorously as pharmaceutical interventions with attention to individual needs, contraindications, and optimization over time. Second, we need better biomarkers, perhaps even point-of-care myokine testing, to personalize and monitor exercise interventions in real time. Can we develop combination strategies that amplify myokine benefits? And critically, how do we translate this knowledge into sustainable behavior change at the population level? From a public health perspective, the evidence is already compelling. Physical activity is one of the most powerful, accessible, and cost-effective cancer prevention strategies available. Thank you for your attention, and I'm happy to take questions.
Shreya Das, are you open for taking questions?
Yes, ma'am.
Okay. Ushali has a question.
Can you please read it out to me?
Yeah, sure. How can we use AI to prescribe a specific exercise dose that releases enough myokines to sensitize the tumor to immunotherapy when patient has been already desensitized to chemo and getting resistance for PARP inhibitors?
So multimodal data streams like the physiological data, biomechanical data, and the different clinical contexts, like as previously stated by her, like the immune to chemotherapies and other therapies, they can be taken into consideration by the AI system, and a digital phenotype must be created based on the risk stratification, the movement competency, as well as the fitness level classification. As a result, using optimization algorithms, the AI can describe like what kind of exercise can be prescribed to her, or any intensity, volume, frequency. As a result, this aligns with the principle of the progressive overload, and this can be monitored with real-time feedback as well as adjustment taken considerably on a continuous basis by the AI system, and it can be updated using the closed loop control systems. For example, like suppose if the performance improves as prescribed by the exercises that given to the patient, then we can increase the load or the complexity or even the intensity if possible. I hope that question answers our question.
Poshali, is it clear? Yeah. Okay, thank you, Shreya. Next is Sanam Preet Kaur. She holds an MSc in Human Genetics from Punjab University, and she will be speaking on AI in early detection of Parkinson's disease. Welcome, Sanam Preet.
Hi, my name is Sanampreet Kaur, and today I'll be talking about how AI is helping us in the early detection of Parkinson's disease before it even becomes clinically visible. I want to begin with a simple but an unsettling idea, which is that Parkinson's does not start at its diagnosis. It starts long before that, years and sometimes decades before that. And by the time we see Parkinson's, we have already missed a large part of it. Before motor symptoms appear, about 60 to 70% of the dopaminergic neurons in substantia nigra are already lost. And all this is happening at a big scale. In 2019, about 8.5 million people were affected by Parkinson's, and this number is expected to be doubled by 2040. So, where are we going wrong? In order to answer this question, let's take a look at the progression of Parkinson's disease. Earlier, it was thought that Parkinson's begins in the motor areas only. But Brath's staging model completely changed that for us. His studies prove that the Parkinson's actually begins in the lower brain regions, like in the brain stem circuits, in the olfactory areas, and gradually it progresses upwards. Its progression is marked by movement of abnormal proteins, alpha synuclein, along the neural circuits. Now, the real question that rises here is, can we detect it before it even becomes visible? And this brings us to the prodromal phase. So, prodromal phase is the initial stage of Parkinson's where the signals are appearing, but the disease is not diagnosable yet. So, the signals may look like reduced sense of smell. There can be REM sleep disorders, there can be constipation and other autonomic disorders, there can be mood and cognition disorders. And among these four, REM sleep disorders stand out the most because of their strong predictive power. According to research, about 70 to 80% of the people who develop REM sleep disorder go on to develop some kind of synucleinopathy later in life. And Parkinson's is one of the most common synucleinopathy. So the signs are not the problem because signs are already there. The problem lies in being able to read those signs. And this brings us to AI. So because the biological signs are subtle and they are diffused and they are distributed across various modalities, clinicians often have problem in discerning them. So the noise is bigger than the signal and there is greater individual variability, and the relationship among individual signs are just too complex to keep track of. But AI thrives in this complexity. Instead of asking, is this signal abnormal? It asks, what pattern emerges when I combine these multiple signals? And what does this look like clinically? Now AI can be used to detect Parkinson's through multiple early markers. And speech analysis is one of the things that AI does. So AI can detect minor changes in the speech patterns like changes in the pitch, changes in the articulation or vocal stability, and it can do that with an accuracy of 90% or above that, something which is not possible for humans. And another early marker is handwriting analysis. So PD people start developing what we call micrographia, which is the progressive shortening of the handwriting. But AI goes beyond that. It can also detect the pressure with which a person writes, the speed of writing, and also the fine motor changes when a person is writing. And gait analysis and facial analysis are also become possible to detect using AI. AI can detect changes in the stride length or the asymmetry of walking patterns. AI can use video data to detect minor changes in the facial expression, because PD people have what we call masked face or hypomimia, in which they have difficulty showing the facial expressions. Now imagine capturing all this data, gathering all this data on daily basis outside the clinician's office, outside the research settings, and this is where the wearables come in. So wearables have sensors attached to them which can detect various types of data in our day-to-day life, like data about movements, speech patterns, and data about gait, and all these data can be integrated into one kind of model. And this is the biggest breakthrough for AI. So each of these signals is useful alone, but it's not enough because Parkinson's itself is not a single signal disease. It is a network problem. So for the first time, our tools are beginning to match the complexity of the problem. So multimodal AI uses multiple biomarkers, voice biomarkers, handwriting biomarkers, and facial expression biomarkers. And then it converges them, which not only improves accuracy, but it also allows us to see what initially was invisible. We are now entering a new phase in how we approach Parkinson's disease. The first step is detection, identifying signals that were already present but were previously unreadable. The next step is prediction, using longitudinal data and computational models to estimate when the disease is likely to emerge. Digital twins is one of the emerging concepts in this field. Now, digital twin is like a computational model of a patient which is built from their own biological and behavioral data. This allows us to simulate disease in a virtual way, so we can track the progression of the disease and we can predict when the disease is likely to emerge, and we can also test interventions virtually. Our final goal is prevention, intervening before any significant neuronal loss happens. While we are not fully there yet, but this is the direction that research is rapidly evolving in. Now, I will leave you with this. AI does not create new information. It is simply revealing the patterns that were always there but were hidden within the complex systems. And if we learn to interpret these signals in time, we may finally move from managing the disease to actually preventing it. Thank you.
Thank you, Sanampreet. That was good. So next we have Ashley Verghese. She comes with a background in microbiology and she will be presenting on AI-guided liposome-based vaccine design.
Good morning. I'm Ashley Verghese. Today I will be presenting on AI guided design of liposome based vaccine carriers for enhanced immunogenicity. This work sits at the intersection of nanotechnology, immunology, and artificial intelligence, aiming to make vaccines more efficient and targeted. When we think about vaccines, we often focus on only on the antigen, but in reality, delivery is equally important. Traditional system faces major challenges like poor antigen stability, limited uptake of immune cells, weak or short-lived immune responses. So now the key question arises is that how can we design a smarter delivery system that protect and enhance immune activation? Liposomes as vaccine carriers. So this is where liposomes comes in. Liposomes are biocompatible vesicles that mimics cell membranes. They can carry both hydrophilic and hydrophobic molecules, making them highly versatile. We can tone the size for lymph node targeting, surface charge for cell interaction, add PEG or ligands for stability and targeting. Because of this flexibility, liposomes are already widely used in modern medicines as well as in drug delivery. However, designing the perfect liposome is not simple. We are dealing with multiple interacting parameters like lipid composition, size and charge, as well as the encapsulation efficiency. And these influence complex biological outcomes like immune cell uptake, antigen release, as well as cytokine activation. Traditionally, this is done by trial and error method, but it is very slow as well as expensive. So this is where the role of AI comes in and it becomes very transformative because AI can learn patterns from previous data, predict how formulation will behave before synthesis, optimize multiple parameters simultaneously. Techniques like random forest and neural network, Bayesian optimization, molecule stimulation allow us to explore thousands of formulation computationally before. So this slide practically shows the workflow and it is quite systematic. We start with data collection followed by feature extraction like lipid properties, then train the machine learning models, predict the optimal formulation and validate experimentally. And importantly, there is a feedback loop that continuously improves the model. So now we look into a case study. For example, in predicting the immunogenicity, we input the liposome size, charge, lipid ratios, and the model predicts the dendritic cell uptake, cytokine production, and T cell responses. This reduces experimental candidates from over 200 to just 10 to 15, saving time and cost. So, looking into the application, this approach has broad application that is mRNA vaccines or cancer immunotherapy, infectious disease, as well as personalized vaccines. So the impact is both clinical as well as translational. Looking into the advantages and limitation, AI offers clear advantages like faster development, reduced cost, as well as better design exploration. But there is limitations as well. Requires high quality data. Models can be hard to interpret, and the experimental validation is still essential. So AI doesn't replace the experiments, it guides them. Looking into the future directions, we are moving towards fully automated AI driven labs, multi scale modeling combining stimulation and ML, digital twin predicting patient response, and importantly, making AI models regulatory ready. So, liposums are powerful but complex vaccine carriers. AI enables faster, smarter optimization. Integration of the computational and experimental workflow is key. AI guided design leads to faster, more targeted, and cost effective vaccines. Thank you.
Thank you, Ashley. Now, next we have Jasmine Steffi, who is currently pursuing her master's in technology in biotech at Karunya Institute. Her presentation focuses on AI in immune system reprogramming. Welcome, Jasmine.
Today, I'll be talking about how we are beginning to reprogram the immune system, not just to treat diseases, but to fundamentally restore balance in human biology. I want to begin with a thought that may sound slightly unsettling. What if the difference between health and disease is not simply the presence of pathogen or even abnormal cells, but how our immune system chooses to respond? Because the same system that protects us with extraordinary precision can fail us in two fundamentally opposite ways. It can remain silent when it should act, or become destructive when it should remain silent. To understand this, we need to move beyond the idea of immune system as a just defense mechanism. It is a dynamic, adaptive networks of cell, signaling pathways and regulatory checkpoints, constantly making decisions. At its core lies immune tolerance, the ability to distinguish self from non-self. But this distinction is not absolute. It is probabilistic, context dependent, and shaped continuously by molecular and environmental signals. And when this delicate balance is disrupted, disease emerges. In cancer, the immune system becomes too tolerant, allowing abnormal cells to survive. In autoimmune diseases, the system becomes overactive, attacking the body itself. So both the conditions arises from the same fundamental problem called immune dysregulation. Let's begin with cancer and see how tumor manipulates the immune system to survive. Under normal conditions, the immune cells such as cytotoxic T cells and natural killer cells identify and eliminate abnormal cells through immune surveillances. However, tumor evolves. Through immunoediting, they pass through elimination, equilibrium, and eventually escape. At this stage, tumor cells evade detection by reducing antigen presentation, altering signaling pathways, and creating an immunosuppressive microenvironment. This is where immunotherapy intervenes. Tumors exploit immune checkpoints like PD-1 to suppress T cells. Drugs like nivolumab block this interaction, restoring immune activity. And even more advanced approach is CAR-T therapy, where a patient's T cells are genetically engineered to target tumor antigen with high precision. But this also introduces a risk like cytokine release syndrome, reminding us that immune reprogramming must be carefully controlled. Moving on to autoimmunity. When the immune restraint fails, the system turns against the body it was built to protect. The immune system does not fail to recognize threats here, but it misidentifies the body itself as a threat. In conditions like rheumatoid arthritis and systemic lupus erythematosus, self antigens trigger immune response, leading to chronic inflammations and tissue damage. Here, the goal is not activation but recalibration. Therapies include immunosuppressive drugs and targeted biologics that inhibit specific inflammatory pathways. However, complete suppression is not ideal. It creates chance for several infections to flare up. So once again, the goal is balance and not the extremes. At this point, the complexity of the immune system becomes a major challenge. That is where AI and computational models play an important role. They allow us to analyze immune interactions, cytokine networks, and patient specific response at a scale that was previously impossible. To conclude, immune system is not just defense mechanism, it is a decision making mechanism. When it functions correctly, we experience health. When it fails, we see disease, either through silence in cancer or through self destruction as in autoimmunity. Reprogramming the immune system is therefore not just about treating the disease, it is about restoring the balance within one of the most complex systems in biology. Perhaps the most important realization is health is not a fixed state, it is a dynamic equilibrium. Thank you.
Thank you, Jasmine. Next, we have with us Shri Deepti, an MSc Biotechnology student at MIT World Peace University. She will be presenting on Immuno-oncology, understanding cancer through immune system.
Hello everyone. I want to start with a question. What if the most powerful cancer fighting weapon in existence wasn't invented in a lab, but was already inside every single one of us? Well, that idea sounds almost too good to be true, right? And for a long time, it was. For decades, cancer treatment focused on destroying cancer cells through surgery, radiation, or chemotherapy. These methods are effective, yes, but they often affect healthy cells as well. We have started to understand how to use the body's own immune system against cancer, a field known as immuno-oncology. As a student from biotechnology background, when I started getting inclined towards cancer research, I kept wondering, why are we always looking for solutions outside the body? Why not work with something we already have? We have a highly sophisticated immune system capable of recognizing, adapting, and protecting us. So the question becomes, can we guide it to fight cancer as well? That curiosity is what makes immuno-oncology so interesting to me. Our immune system can detect abnormal cells, but cancer, well, it is a master of disguise, right? And is also highly adaptive. It develops ways to hide and suppress immune responses. So the issue is not the immune system is weak, but that cancer is able to escape it. Immuno-oncology works by correcting this imbalance. For instance, checkpoint inhibitors that remove the brakes on immune cells. A widely used example is pembrolizumab. CAR T cell therapy modifies a patient's own immune cells to better recognize cancer. And cancer vaccines and monoclonal antibodies help stimulate or direct immune responses. A major part of this progress comes from biotech startups, with innovations in cell-based therapies, genetic engineering, mRNA platforms, and immune profiling. And some of the notable startups in this field are Kite Pharma, which helped bring CAR T therapy into real clinical use by engineering a patient's own T cell to recognize and attack cancer. These modified cells are reinfused into the body, acting as living treatments, and have shown strong results in certain blood cancers like lymphoma. Juno Therapeutics expanded this approach by working on both CAR-T and TCR-based therapies, allowing them to target a wide range of cancers, including solid tumors, while also improving the safety and effectiveness of engineering immune cells. Next comes Moderna, which is developing personalized cancer vaccines using mRNA technology, where vaccines are designed based on individual tumor mutations to help the immune system recognize and respond more precisely to cancer cells. Next is adaptive biotechnology, which is mapping immune responses using large-scale sequencing of T cell and B cell receptors, helping researchers understand how the immune system recognizes cancer and identify new targets for therapy. Next is Iovance Biotherapeutics, which developed tumor infiltrating lymphocyte therapy by extracting immune cells already present inside tumors, expanding them in the lab and reinfusing them to strengthen the body's natural anti-tumor response. Next is Spade Therapeutics, which is working on stem cell derived immune therapies that can be produced in advance and used off the shelf, which aims to make immunotherapy more scalable and accessible. Next is Allogene Therapeutics, which focuses on donor-derived CAR T therapies, which reduce the need for patient-specific cell preparation, helping lower treatment time and cost while improving accessibility. And next comes Agonist, which works on cold tumors. So cold tumors are the ones that do not respond well to existing immunotherapies. So developing treatments that can activate immune responses and make these cancers more treatable. When we look at real world outcomes, the impact becomes clear. Take advanced melanoma, for example. A decade ago, outcomes were limited. Today, with therapies like nivolumab, patients are seeing long-term remission. One well-known example is Jimmy Carter, former US president. At age 90, he was diagnosed with metastatic melanoma that had spread to his brain. After treatment with pembrolizumab, his tumors disappeared and he lived for many more years. CAR T therapy has also shown strong results in certain blood cancers, especially where other treatments failed. This shows not just effectiveness, but new possibilities. When I reflect on my initial question, why not use our own immune system? Immuno-oncology feels like one way of answering that. It connects research, innovation, and patient outcomes and continues to evolve as we learn more. To conclude, immuno-oncology represents a shift in how cancer is being approached, from directly targeting the disease to working with the body's own systems. The future of immuno-oncology is moving towards treatments that are not just personalized but predictive, with the convergence of AI-guided drug discovery, personalized mRNA vaccines, and next-generation cell therapies where we can identify risk, intervene early, and design therapies tailored to each individual's immune system. As a young researcher, what stands out to me is that this field is not just expanding treatment options, it is changing how we understand the relationship between the body and disease. Thank you.
Thank you, Sridipti. So, do we have any questions for her? I'll give it some time, like I'll also check the YouTube live. If there is any question for Sridipti. Poshali has a lot of questions which can be answered sometime later. But Poshali, do you have any question for her? If not, let's proceed in 30 seconds. Let's just wait. Okay, so no questions for Deepti. Let's move on to the next speaker. She is Kompala Jhansi, a bioinformatics student at Savita School of Engineering. She'll be discussing about atrophy crisis in professional sports. Welcome, Jhansi.
Hello everyone, I'm Jansi. I'm currently a final year engineering undergrad student specializing in bioinformatics. Today, I'll be discussing an emerging biological and technological challenge in allied sports, which is in season sarcopenia, and how the AI driven peptide discovery targeting myostatin offers a precise, safer, and future ready solution. To understand why this problem matters, we first need to look at what's actually happening inside an athlete's body during a competitive season. Elite athletes compete under high density mat schedules, often playing multiple games per week with limited recovery time. Surprisingly, this environment leads to in season sarcopenia, which is a measurable loss of lean muscle mass despite peak level training and nutrition. The current recovery strategies are largely passive, like taking rest, ice baths, compression, and maintaining a proper healthy diet. While these are helpful, but they do not actively prevent muscle degradation at the molecular level. So, to sustain the performance across an entire season, we need proactive muscle preservation, not just post damage recovery. So, if muscle loss is occurring even in elite, the key question becomes what biological mechanism is driving this breakdown. It is myostatin, which is also known as GDF8, growth differentiation factor 8, is a member of TGF beta superfamily. It functions as a negative regulator of skeletal muscle growth, essentially acting as a biological brake. When myostatin signaling is elevated, muscle hypertrophy is suppressed even under the intense training stimuli. Therapeutically, inhibiting myostatin offers a way to preserve or stabilize the muscle mass without relying on systemic hormonal manipulations. However, identifying myostatin as the target is only part of the solution. The real challenge is how to inhibit it safely and selectively, and this is where AI becomes transformative. Traditional drug discovery is slow, expensive, and often inefficient. In this approach, AI and machine learning accelerate the peptide discovery by analyzing vast biological datasets. The workflow begins with compiling multiple millions of natural peptide sequences, and then machine learning models predict which peptides show high affinity binding to the myostatin prodomain. The docking simulations then visualize structural interactions and binding stability. From this process, only the most selective and stable peptide candidates are then shortlisted. The pipeline dramatically reduces development time, cost, and off target risks. What's important here is not just the speed, it's precision. And the precision fundamentally challenges how we think about performance intervention. Traditional systemic steroids operate broadly, affecting multiple tissues and pathways. They carry high toxicity, long term health risks, and violate WADA anti doping regulations. In contrast, AI designed peptides offer target specific molecular modulations. They often minimize off target effects, improving safety, and have the potential for regulatory compliance. This represents a shift from brute force enhancement to precision performance biology. That precision is critical because myostatin doesn't act alone. It belongs to a highly interconnected protein family. Myostatin shares structural similarity with other TGF beta proteins such as GDF11. Non-selective inhibition could be unintentionally affect cardiac, neural, or metabolic pathways. So, the AI-guided structural modeling ensures that peptides bind only to myostatin, avoiding the related proteins. This selectivity is essential for both clinical safety and the translational success. Once selectivity is achieved at the molecular level, the next step is ensuring safety and efficiency in real athletes. Therapy effectiveness is monitored using non invasive biomarkers, particularly the muscle protein synthesis rate. This allows real time, data driven dose adjustments. The result is athlete specific optimization rather than using one size fits all protocols. This approach maximizes benefit while minimizing the risk. What makes this approach especially compelling is the same biology driving athlete performance is also central to aging. Elite athletes serves as high-stringy validation cohort for safety and efficacy. Once validated, the myostatin inhibition strategies can be extended to sarcopenia, cachexia, and age-related muscle loss. This opens pathways into longevity and health aging market far beyond the sports medicine. As science advances, however, it also challenges the existing regulatory frameworks. Then, AI optimized bioidentical peptides blur the traditional doping definitions. Regulatory bodies must distinguish between the nutritional support, therapeutic intervention, and pharmacological enhancement. Future WDA policies will need to evolve to address advanced biotherapeutics. Since the science is moving faster than policy, proactive regulation is essential. For both the regulators and clinicians, mechanistic proof is very crucial. The techniques such as molecular docking and cryo-electron microscopy confirms the peptide myostatin binding, and the structural data shows that these peptides prevent myostatin from activating its receptor. This provides direct molecular validation of the mechanism of action. With the mechanism confirmed, the final question becomes how do we implement this in real competition? The protocol begins with pre-season baseline assessments and the microdosing. Early in season phases integrate therapy with biomarker feedback, and the peak performance phase focuses on rapid recovery and the load optimization. Late season strategies emphasizes injury prevention and muscle preservation. The result is sustained performance with reduced muscle loss. When all of these elements come together, we arrive at a new model of performance care. In conclusion, AI driven myostatin inhibitors represents a paradigm shift in sports medicine from moving reactive recovery to precision molecular performance preservation, with implications extending into aging and longevity medicine. Thank you.
Thank you, Jansi. Now we have Venkateshwaran TV. He has completed his master's in biotechnology and is currently working in computational biology in rare diseases. His presentation focuses on AI in rare disease genomics for precision mental health. Welcome, Venkat.
Yeah, hello all. Thank you very much, Sukant. I hope all of you can see my screen. So as said by Zenith earlier, I am focusing on AI in rare disease genomics for precision mental health medicine. So basically, I'll be bridging the sequencing data and clinical insight ideas. Uh, so the main problem here is in the mental health disorders is the heterogeneity and the high complexity of those disorders. And genetic causes one of them. It's not completely genetic. Mental health disorders are caused by a plethora of factors. They can include environmental factors and genetic factors as well. And so today will be focusing on the genetic factors here. Um, the other issue is traditional diagnostic, which is actually slow and it's not reliable to identify or to diagnose these disorders. Uh, so in order to study the genetic causes of various mental health disorders, scientists and a lot of companies they use genomic data or to be specific, they do sequencing, like whole genome sequencing or whole exome sequencing. And in order to identify copy number variations, they look into chromosomal microarray data or even MLPA data. And the main bottleneck, the advantage of the sequencing is get all the uh, you get a lot of data, you have a massive data set, but the problem is interpretability. So due to this interpretability problem, this is where AI steps in order to enhance the genomic data analysis. So one of them is machine learning. So labs basically use random forest so that these AI models can prioritize pathogenic or likely pathogenic variants. They use deep learning where it can find the variant effects, like what it causes, whether it's a stop loss or whether it's a null variant and all. And they also use natural language processing which basically, which to simply say, it correlates or integrates the clinical record with genomic data. So what happens here is, so by the labs, genomics lab usually have bioinformatics pipeline before the analysis of the data starts. So they use different tools like GATK to identify the variants, and there are annotation tools like ANOVAR or VEP or SNPF, which annotates the variants. And here there is this machine learning based integration, which enhances the process of variant prioritization, which will then be useful, and which will then can catalyze the process of variant analysis and also the report making process, because the final result is the report which gets delivered to the clinician. And also, apart from genomics, transcriptomics and proteomics should also be looked into in order to find out what a particular gene variant causes to a certain disease. So AI integrates all these data, genomics, transcriptomics, and proteomics, where you have the variants, the rare variants identified, and transcriptomics data can give you the gene expression pattern, and you can also, the proteomics and all can give you the metabolic pathways here and different protein interactions. So these are actually integrated by AI. And when we talk into application to mental health, it can be useful to study the genetic causes of autism or attention deficit hyperactivity disorders. There are a lot of subtypes of intellectual developmental disorders, and also other rare neurodevelopmental syndromes or schizophrenia. And so flow is, it starts from identifying the particular variant, and it goes till the mechanism where AI, after finding the mutation, the algorithms can map to different pathways. It can be a mitochondrial distress mechanism, or it can even be a synaptic dysfunction mechanism, and it can also predict the impact of the variants. So all this can lead towards precision medicine, where you can have targeted therapies based on each of the patient's genetic profile. And the major challenge here is data bias, because if you see in genomics, the data is always biased towards certain populations, like especially the European populations and all. But when it comes to the Asian populations or even the Middle East population, the studies of these variants or these particular disorders are comparatively less. And there are like interpretability issues, since AI works like a black box model. The thing is, yes, it can prioritize pathogenic variants as said earlier, but the thing is how it is done, what reasons are used, those are not clear. And in order to take a clinical decision, you should require an explanation. So that is slightly vague in this case. And there are many ethical issues, which basically, if you want me to quote an example, I can say that there can be data misuse here. So that is another disadvantage. And in the future directions, scientists are going towards looking into more explainable AI, so that they can mitigate the interpretability issues. And people have to expand the global databases, and clinical integration is the need of the hour, so that a proper integration can make it clinically sensible. So this is the end of my presentation. So I'm open to any questions.
Any one of you, if you have a question, please type down in the chat box. Venkat, we can wait for a few minutes.
Yeah.
Venkat, you can stop your screen.
Okay, so there's no question. Let's move on to our next speaker. Our next speaker is Poshali Bose. She has a master's in biophysics, molecular biology, and bioinformatics. She will be exploring AI and gut microbiome in metabolic disease detection. Welcome, Poshali.
Hello everybody. Today I'm going to talk about how AI is actually can help us in early detection of the metabolic diseases. So basically metabolic disease like obesity, type 2 diabetes, insulin resistance and non-alcoholic fatty liver disease are among most pressing global health challenges at this point. Even though one of the biggest limitation is that in managing these conditions because they are diagnosed only after significant metabolic damage has already been caused. So which raises a critical question. What if we could detect these diseases early at a stage where the intervention is still highly effective? So, in recent years, the gut microbiome has emerged as a key player in metabolic health, with growing evidence suggesting that microbial enzymes mostly, rather than just microbial composition, serve as direct functional mediators of host metabolism by regulating essential processes. So what I'm going to talk about today on this topic is that we are going to talk about the challenges and the microbiome's role. So the limitations of the current methods that is available at this point, and also how AI is actually solving the problem, and also we'll give a little bit emphasis on the different methodologies, how they can be used in overcoming the problem, and what can be the impact and future in order to overcome the issue. So basically, we know that there are different types of metabolic diseases like obesity, type 2 diabetes, insulin resistance, NAFLD. Not but so this is not the only list. There are a lot of diseases whose causes are not even known to us, but we know that gut microbiome play essential role by maintaining the brain axis. A lot of diseases, their early detection and their preventions can be done by the scientists. So at this point, the major limitation is actually the late diagnosis, which happens only after significant damage has been caused. So it takes like a lot of effort to restore the gut microbiome health. But just because we are not aware of most of the bacteria available in the gut and their history and their functions are not known to people, and it's the least unexplored part of the science, so it has been very hard, and which raises a critical question like how can we actually know more about these gut organisms and get the benefits out of this and how it can actually influence the health of human being. So early intervention is crucial for effective management and the better outcomes. So, what is the central role that is played by the microbiome? So, basically, there are a lot of like a lot of bacteria are present in our gut, and they are performing different functions, and they are the key player of the metabolic health. So microbial enzymes, it's just not about the composition, but also the function as well, because the population of different bacteria can literally alter the population of the other bacteria. So based on that, the whole metabolism can be changed. So, and also the microbiome has a very unique environment, which causes the population to vary based on different species, different individuals of the same species, based on their eating habits, and a lot of other factors. So basically, there are a lot of pathways involved in it, like SCFA production, bile acid transformation, lipid metabolism. So somehow, if these pathways are not balanced, that can cause high high inflammation in the body. And we know that the high inflammation not only causes this disease, but this type 2 diabetes, um, metabolic diseases, but it is also one of the reasons for the autoimmune diseases as well. So basically, so out of a lot of examples of this, I'm giving couple of examples. For example, like enzyme from if Groundnut and our interstitial release promote gut health by producing butyrate. Also bile mostly, which is actually um, metabolized in the liver, but also like it has a lot of impact on the production of the bilirubin and biliverdin. So from lactobacillus and bacteroids, they regulate this bile acid signaling, and we know how their composition can actually impact the whole pathway, and thus impacting the health of the individual. Also from gram-negative bacteria, which causes inflammation and insulin resistance due to their polysaccharide. Other than that, Clostridium species produces TMLIS, like TMAO, with the help of this TMLIS, which is linked to the fatty liver. And we know that at this point, non-alcoholic fatty liver has been an epidemic to mostly the third world countries. An anti-inflammatory effect can also be generated by the population of the gut microbiome. So reduced biter rates actually leads to improved anti-inflammatory signal, and other than that, all this at the end lead to the improved insulin sensitivity as well. So, what are the limitations of the traditional methods, and what traditional methods are currently available? So, at this point, there are different traditional methods available, like UPCR, which uses 16S RNA as a standard one. Other than that, different types of gel hybridization protocol, fish methods, hybridization, RFLP, all these methods are actually available, but these methods have their own drawback. So they themselves are not capable of solving the issue. So basically, traditional methods have been essential in gut microbiome research, so providing the initial insights into the composition and the diversity. So, for example, the PCR, which enables the targeted gene amplification and limited by primer bias and inability to detect unknown taxa, while denaturing gradient gel electrophoresis and TRFLP help in getting the fingerprinting of the community, but it suffers from low resolution and reproducibility issues because of the accurate species identification, lacking of accurate species identification. Other than that, fish allows spatial visualization of microbes within their environment, yet it is constrained by probe specificity and limited scalability. Other than that, the introduction of the 16S RNA gene sequencing has revolutionized the field by enabling high throughput taxonomic profile, but it produces only compositional data, lacks functional insights, and is affected by methodological biases. Similarly, other than that, shotgun metagenomics, which is a famous way to get all these informations in the modern era, it also has its own drawback because it is computationally very intensive and prone to assembly errors and cannot reliably distinguish active metabolic processes. Overall, these traditional approaches provide static, descriptive snapshots of the microbiome and fail to capture the dynamic nature of it. So, how does the structure and function relationship and how the dynamics of the enzyme is actually controlling the behavior of the enzyme and its function. So this enzyme level mechanism highlights the need for more integrative and advanced analytical strategies. So even the advanced omics, which includes the metatranscriptomics and metaproteomics, it introduces a lot of data heterogeneity, but it itself suffered from heavy noise, difficulty in linking the gene to active processes because of the experimental variability leading to inconsistencies. So it also lacks a standardization procedure. Additionally, low abundance molecules distinguishing host from microbial signals, accurate annotation of the metabolites are still lacking. So these approaches are computationally intensive as well, also need highly advanced bioinformatics expertise. So importantly, most multiomics data reveal correlation rather than the causation, so limiting the mechanistic understanding or the dynamics of the current state of the gut microbiome, with the link to their function or the other pathway. So basically, all these problems can be overcome at this point by the help of the AI. So the biasness is there because of the sampling, sensitivity issue, resolution issue, and then these methods are fail to capture the function and is actually capturing only the static view. So this is making the things complicated. So this can be overcome by the AI because AI is actually integrating all these data from the large scale multiomics and as well as the shotgun sequencing, enabling insights which was actually impossible to be done by the traditional methods, bridging the gap between the microbial identity and functional activity. It also transforming descriptive science into predictive and mechanistic learning. So basically, AI is actually have different topics. So AI has different parts. So we know that AI is enabling the integration of interpretation of complex microbiome and multiomics data sets. So within AI, the machine learning focuses on learning patterns from data, while deep learning is using the multi-layered neural network to automatically extract complex features, especially from large-scale genomic and sequencing data. In supervised learning, models are trained and label datasets. So where the healthy and disease sample are collected and then the statistical analysis is being performed. For example, random forest analysis, which includes the robustness and can rank the important features, making it ideal for making it actually ideal for to know the microbial enzymes, how is it linked to the obesity and diabetes. Then support vector machine are effective for high dimensional microbiome data, enabling precise classification of disease state like XGBoost. Also help in a gradient boosting model, which is actually improved prediction accuracy and is commonly used for biomarker discovery and risk prediction. And in unsupervised learning, no labels are used, which by using this CAPE means clustering, uh, microbial similarity, helping identifying the disease associated with microbiome profiles, and hierarchical clustering also helps in getting the relationship between the microbial species and enzyme across the sample. So this and also the dimensionality reduction can be performed by PCA and TSNE like methods, which helps in getting the more precise data. Other than that, there are deep learning models like say, different neural networks models are available which capture the sequential time series microbiome data, which helps in studying the disease progression. And other than that, advanced neural network models also help in getting the network and the pathway and how these different pathways are connected to each other. And then, with the help of the Bayesian networks, ultimate prediction and how they are connected to each other and the pathways and the causing microorganisms can also be predicted. So basically, AI is not only helping us in enzyme function prediction, but it can also give shed light on the pathways as well. So there are different deep learning models like CNN RNNs, as I have already talked about, predict the enzyme function, identify the hidden pattern mostly beyond the motif level, which helps in understanding the function potential, crucial for linking the microbial genes. Also, it helps in biomarker identification by reducing the dimensionality. So, gives us the biological meaningful variation for clearer insights. The supervised model includes random forest SVM, and it actually help in identifying the biomarkers. And also, AI is uncovering the hidden patterns and interactions by doing the unsupervised learning, and then it identifies the microbial community and its subtypes and disease association with it. So it integrates enzyme, metabolites, and host factor to know the cause, to understand the cause of the disease. So AI is helping in enzyme prediction, biomarker discovery, pathway reconstruction, and causal relationship. So what is the impact of AI in precision medicine? It enhances sensitivity. It is enhancing the novel targets as well, shedding lights to the new pathways, which definitely needs to be validated by the experiments, but it still can give us a lot of information about the pathways that have completely been unexplored before. And other than that, it helps in modulation, personalize the medicinal approach. So every individual has different gut microenvironment, so it gives the opportunity to do the personal medicine as well. Proactively, it is transforming the healthcare and provides a deeper mechanistic insights into the host microbial relationship. But it also has challenges because it remains a significant hurdle for because of the interpretation of the model needs more standardization. Rigorous experiments are needed, but and as a future direction, it's still helpful in a sense that it is rapidly addressing the interpretation. It's advancing the system biology and improving data integration and validation. And rapidly, it's actually transforming the research into the professional healthcare, professionally at health level, where the disease can be predicted early, and human life um, could be improved. So, what is the future of this metabolic health? So, we are at the descriptive stage at this point. So, the AI can help us to predict the biomarkers so that we can get a personalized prevention in future. Thank you.
Thank you, Poshali. Now, you have been very active and asked a lot of questions, which is quite good. Now, let's see if you receive any questions. We can give it a minute. Please type your questions if you have. So there's no question for Poshali at the moment. Let us move to our next speaker, who is Sakshi Sharma. So she has a background in microbiology, and she will be presenting on tracking resistance gene evolution using AI. Welcome, Sakshi.
Good morning, everyone. I am Sakshi Sharma, a postgraduate in microbiology, and today I'll be talking about how AI and genomic surveillance can help us track antimicrobial resistance in real time. This topic sits at the intersection of clinical microbiology, public health, and data science. The key question here that we are going to explore is, can we move from reacting to resistance to predicting it? Antimicrobial resistance, or what is widely known as AMR, is often called a silent pandemic. Currently, it has caused about 1.27 million deaths directly and contributes to nearly 5 million deaths annually. By 2050, AMR, or antimicrobial resistance, could surpass diseases like cancer as a leading cause of death. The real issue is resistance genes evolve rapidly and they spread before even we detect them. So, the challenge is not if AMR will worsen, it is rather how fast we can respond. Current diagnostics and surveillance systems are too slow. Culture based testing can take days to weeks. Genomic analysis is often retrospective, meaning we study outbreaks after they have happened. Even tiny mutations, like single nucleotide changes, can cause resistance but are often missed. This delay creates a critical window where resistant strains spread unchecked. Now, this is where AI comes in. With whole genome sequencing, we generate massive microbial datasets. AI and machine learning helps convert this data into real-time insights. Specifically, AI can detect known and novel resistant genes, identify mutation patterns, also predict future resistance variants, and provide early warnings. So, instead of reacting, we start predicting resistance before it becomes visible clinically. Now, this slide here shows the pipeline. First of all, we collect patient samples and perform whole genome sequencing. Then, AI models that are basically used to detect mutations, flag high risk variants, and these insights are converted into the clinical alerts. The key advantage here is what used to take weeks now takes hours. So, this enables faster decision making in clinical settings and hospitals. Now, let's look at a real world scenario. Let's say a patient sample is routinely sequenced. Now, AI detects a subtle mutation in a resistance gene. Even before lab confirmation, the model predicts resistance to a common antibiotic, and the clinician here is already alerted at an early stage. This will allow an immediate treatment adjustment, infection control measures. One mutation, if missed, it can cause a ward level outbreak in days, which is why it matters a lot. AI driven surveillance, it impacts multiple levels such as faster treatment, doctors can switch antibiotics early, better infection control, prevent spread before the outbreak occurs, improved antibiotic stewardship, and also helps in smarter prescribing policies. Overall, it improves both patient outcomes as well as the efficiency of the hospitals. The future vision is global, integrated surveillance, not just hospitals, but also targeting wastewater, environment, regional data sets. So AI can track resistance across border in real time. For example, wastewater can act as an early warning system, and this creates a global AMR intelligence network. Despite the promise, there are major challenges which are as follows. First is incomplete data sets. Many regions they lack sequencing infrastructure, and another one can be lack of standardization, like data sharing requires uniform protocols. Another factor is clinical trust. AI predictions must be validated and accepted by the clinicians. So, without solving these challenges, scaling AI will be difficult. AI is not here to replace the microbiologists. We need to understand that. Instead, AI has come into picture in order to enhance their ability to act at an early stage in order to prevent the widespread. And the goal here is very simple. First is to detect, like identify the resistance at an early stage. Then comes prediction or predict, where we understand the future mutations. Then the most important one, act, which makes faster the clinical decisions. So if it is all implemented well, AI can shift us from reaction to prevention in AMR real quick. Thank you so much for your patient listening.
Thank you, Sakshi. Now, there was a question by Poshali for Deepti. Deepti would like to answer that. Deepti, I can read the question to you.
Yes, ma'am.
Okay. So the question is, docking simulations have drawback in simulating nucleic acid molecules, especially like modified RNA. But nucleic acid-based drugs by conjugation has been a hot ongoing research. So how AI can help in bridging this gap?
Okay. So from what I understand, traditional docking simulations do have limitations when it comes to modified RNA. Maybe because of their structures, they are like complex. So AI can help, for example, by using machine learning and molecular dynamics approaches as well. So, which can help to predict in a better way how these molecules behave in real biological environments. So, when I talk in the context of immuno-oncology, as I mentioned, Moderna, which is working on mRNA-based vaccines, and also other mRNA-based therapies. So, machine learning and these dynamic approaches, molecular dynamic approaches, this becomes important for designing more effective and stable delivery systems. So, yeah, AI helps bridge that gap by improving prediction accuracy and guiding experimental design. Even though it wouldn't completely replace lab validation.
Okay, thank you. Beautifully answered. I hope that answers you, Poshali. Okay, so now we move to the final presentation, and I'm pleased to introduce Dr. Alfredo Gezzi from the Department of Biology at the University of Puerto Rico. Dr. Gezzi's research focuses on neurobiology, behavior, and the molecular mechanisms underlying addiction and adaptation. So in this session, he will be presenting on epigenetic mechanisms of alcohol neuroadaptation in Drosophila. Let us move to the presentation.
Hi, my name is Alfredo Gezzi, and today I will tell you about the work that we do in my lab at the University of Puerto Rico, Rio Piedras campus, in which we're trying to understand the epigenetic mechanisms of alcohol neuroadaptation in Drosophila. So my lab is interested in the mechanisms of neuroadaptation, and this means how does the brain adapt to different experiences, whether it's exposure to drugs or to high stress, and how those changes lead to changes in behavior, such as drug addiction or sometimes homeostatic dysregulation. In essence, what we're trying to answer is how does the brain adapt to an ever-changing environment, and what are the consequences of these changes? So everybody knows that the brain, whether it's a human brain, a mouse brain, or the tiny fly brain, is one of the most adaptive organs in an organism, right? It has this capacity to change its structure and its function over a lifetime in reaction to environmental diversity. So one of the key elements that makes this neuroplasticity work is that there's a highly controlled balance between excitation and inhibition of neural activity, in which when the brain is too active, there's mechanisms that try to compensate for that activity to bring it down to normal. And if it's too low in activity, there's mechanisms to bring it back up to the normal activity. In essence, these homeostatic mechanisms prevent the brain from going to extremes such as a coma or a seizure, essentially. Right? So using this framework, people have postulated theories about how drug dependence works. And this theory revolves around this idea that the balance of neural activity between excitation and inhibition is disrupted whenever an organism is exposed to a drug that has psychoactive effects, such as, for instance, alcohol or some opioids, right? Like alcohol, for instance, is a sedative, so it could push the brain activity to a more sedative state. In response, the brain may trigger adaptations to push this effect of the drug to the opposite side in terms of opposing the effects of the drug, right? However, once the drug is metabolized and excreted from the organism, you are left with the adaptation, which usually it's expressed in the organisms as the withdrawal state, in which now you see the opposite effect of the drug as a more hyperactive nervous system. The interesting thing about this is that one of the easiest ways to restore the original balance is to go back to the drug. So this will be known as the dependent state. So this cycle, it's actually repeated every time a user uses a drug and perpetuates this type of adaptation, right? So in my lab, we use a Drosophila model to study how does this adaptation work, okay? And in essence, we use Drosophila because not only has a very accessible genome that allows us to do very rapid mutant analysis, we can test the effects of different genes, and we can manipulate those genes in a very effective way, but fruit flies show alcohol responses that closely resemble those in humans. They show tolerance, which is the initial resistance to the drug, and they show alcohol withdrawal, which is the symptoms that you get once the drug is metabolized. And we can easily quantify these behaviors using this model. For instance, we can put flies in vials and pump these vials with alcohol vapor and see the effects of the alcohol in the behavior of the flies. In essence, when we start to expose the flies to ethanol vapor, you will see that they will knock down to the bottom of the vial rapidly as they become intoxicated. However, once you turn the alcohol off and then they get just fresh air, they start to recover within a time frame of 30 minutes to 1 hour. So this is a very rapid assay in which we can quantify how fast and how fast they knock down and how fast they recover from this alcohol exposure. If we repeat this assay on a second time, essentially 24 hours later, we can see that flies that are recovering from the second exposure actually recover faster than flies that are undergoing the first exposure. So this shift in the recovery curves that you see here on the bottom left to the left indicates that the flies are becoming adapted to the presence of alcohol and now are recovering faster. You can express this in the recovery curves as shown here, or as average recovery time between the first and second exposure. You see a significant diminishment in the recovery time in the second exposure. This can also be expressed as a tolerance index, in which you subtract the second exposure from the first exposure and calculate the difference, normalized to the first exposure, and you come up with an index. So in essence, it is really easy to see flies becoming tolerant to alcohol. Similarly, and I'm not going to go into much of the details of how we do this, but you can see symptoms of withdrawals of flies that have undergone a first exposure to alcohol. In essence, when we stimulate the brains of a fly with a electroconvulsive shock, we see that flies will start ceasing much faster than flies that have had alcohol will start ceasing much faster than flies that have not had alcohol in a prior instance. So these two elements shows us that we can easily quantify not only tolerance to alcohol, but also the appearance of withdrawal seizures. So over many years, we have identified many genes that are responsible for these adaptations. One of which is this BK channel that is encoded by the genes logo. This BK channel is a large conductant calcium-activated potassium channel that is involved in regulating synaptic excitability and neurotransmitter release. So the model that we had, at least until the last decade, is that under normal conditions, there's enough of the presence of this channel in the membrane of the presynaptic terminals that will mediate the proper fusion of vesicles to sort of modulate a basal capacity of firing of neurons. However, in the presence of alcohol, where the activity of the neuron is suppressed by the sedative effect of alcohol, then we don't see a basal capacity for firing anymore. So the nervous system is forced to produce more of these PK channels to enhance the capacity for firing. However, now that the alcohol is gone in the withdrawal, then the increased presence of the PK channels gives rise to an increased capacity for repairing firing. However, over the last few years, we have, of course, discovered that it's not only a single gene that participates in the process, but it's a network of genes that whenever an animal is exposed to alcohol, they turn on and can enhance tolerance as well. So our research objectives is try to identify more of these genes that are important in the adaptation to alcohol, and also how these genes affect the physiology of the nervous system, and of course how are these genes regulated. And in this specific question, we have actually made some recent advances that are very encouraging into trying to solve this potential problem of organisms becoming adapted to drugs such as alcohol. So the answer to the short answer to this question is that many of this regulation lies into the field of epigenetics. Okay, so for those of you that don't know exactly what epigenetics means, it's the structural adaptation of chromosomal regions that register, signal, and perpetuate altered activity gene states. In essence, epigenetics are modifications that happen on the chromatin that can change the structure of the chromosome, and these changes in structure can allow for transcription to go on because it makes the DNA accessible to transcription factors and the RNA polymerase that in essence end up turning on the transcription of specific genes. So we have quantified the modifications that happen across the genome in flies after alcohol exposure and seeing if this correlates with specific changes in gene expression of genes that are important for alcohol tolerance. And many of the genes that we have found have peaks of acetylation at their promoters after there's an exposure to alcohol. Okay, so the next question was, who are the enzymes or the most plausible candidates for taking care of these modifications? And we know that many of these modifications are carried on by specific enzymes such as histone acetyltransferases or HATs depicted here that are the ones responsible for adding acetylation marks into the chromatin. Similarly, there's HDACs, which are the histone deacetylases that can take those marks from the chromatin and lead to a more compressed state. So by studying the potential enzymes that are involved in this process, we can see if there's specific genes or enzymes that can be modulating this process. So far, we have found that there's at least 2 alcohol-responsive histone acetyltransferases that mediate this process. The first one is TIP60, which is the Drosophila homolog of the human histone acetyltransferase CAT5. And the second one is MEGIDI, the Drosophila homolog of the human CREB-binding protein CBP. As code readers, these 2 enzymes recognize the epigenetic codes for initiating transcription, and they have been shown to play critical roles in the activity-dependent responses by coupling chromatin remodeling to transcription factor recognition. So our current working model is that transcription factors yet to be determined associated with different hats, such as TIP60 and MEGIDI, to acetylate the promoters of alcohol-responsive genes, and this action of acetylation promotes their transcription to lead to tolerance and withdrawal phenotypes. So in essence, this is where we're at now, right now. We're trying to decipher what are the genes that are being modulated by each of these 2 different enzymes, and what are the consequences of their action in different aspects of the alcohol response phenotype. So many questions remain. How do different alcohol-induced genes affect synaptic transmission? We still have to determine that. Which are the circuits in the brain that are most important for regulated alcohol-related behaviors? And how does alcohol neuroadaptation interact with other homeostatic processes in the brain that can explain some of the side effects of alcohol tolerance and withdrawal? So with that, I just want to thank the people in my lab that help in this work. We have a large number of undergraduates and graduate students that participate in this work, not all of which are mentioned here, but I just want to make sure that the work that is being done by these people is recognized. We, of course, have funding from different sources, including the University of Puerto Rico, the NIH, and the NFL. So thank you for your attention, and please reach out if you have any questions about this topic or any related areas of research. Thank you.
Thank you, Professor Alfredo. Now, Venkat has a question for Sakshi, which can be answered by Sakshi tomorrow or later. Now, I have an announcement to make. Those of you watching us on YouTube Live, we have a feedback form over there on the comment box. Please go ahead and fill that. We have also posted that same feedback form over here in the chat box. Please copy that, fill the form, and send to us. I'll give it a minute. You can copy the link and save it. You can fill it later. Okay. So this brings us to the end of day 2. Today's sessions spanned a wide range of domains from brain science, cancer, AI systems to healthcare, sustainability, and advanced biological applications. We look forward to continuing these discussions in day 3, which is tomorrow. And thank you everyone for being a part of the conference. Thank you.